There's quite a number of chairs in the front rows which are not taken yet. So, good morning everyone. I guess it's a good sign that we are starting late because it tells us the registration took longer and there's quite a number of people. So we've actually got more than 350 registrants for the meeting, which is record. So that's great. Let me start out thanking you for being here. I would like to welcome especially our Scientific Advisory Board. As most of you know, we do the HIPS Symposium in connection with the meeting of the Scientific Advisory Board, and they have helped us establishing HIPS. It's now HIPS in its 15th year. Which is something we're probably going to celebrate next year with a two-day symposium, but that is to be put into your calendar soon. Let me also thank the supporters here at Saarland University, our friends at the Helmholtz Center in Braunschweig, for being here partly and supporting us over the years. I'll try to give you a quick update of what has happened in the last year, and then we can get started into the scientific program, which I'm looking forward to. For the guests who hear about HZI and HIPS for the first time, we do have a few slides. The Helmholtz Center in Braunschweig is sometimes difficult to connect to, as you can see here. And when we got reviewed, which is now exactly 15 years ago, this was, as long as this is not working, I'll tell you some stories, right? So this was one of the big questions, whether it would ever work over that distance of more than 500 kilometers to establish a site of the center in Braunschweig. And I believe we've proven the people who were critical wrong over the years. Of course, for us, it's always been equally important to closely interact with the local university, which helped us to start the HIPS. I trust this is going to work. Otherwise, I will tell you more stories. We have started this institute a while ago, the center in Braunschweig. I need the map now. Is in the northeast of Germany, 500 kilometers from here. And the center has expanded quite a bit over recent years. The center now has approximately 1,000 people, almost 300 of which are located here at the Helmholtz Institute. And the center has three major research foci: host pathogen and anti-infectives, and the HIPS is driving the anti-infectives part, but there's also a couple of colleagues in Braunschweig who are part of it. HIPS is still, and that's our USP, the only research institute in Germany which has pharmaceutical research as its main topic, and these are the PIs, in the name of which I would like to welcome everybody again. We've expanded the number of PIs. There are some PIs on the Braunschweig side. I think we have most expanded our bioinformatics part. You'll get to know these people when you are here. They are all in the audience, and we closely interact with the site in Braunschweig in this topic, anti-infectives. And our major foci are medchem, drug delivery, bioinformatics, and natural products. And we have sessions covering aspects of all of these as of today. So what we basically do, starting from medchem or natural products, we get chemical matter, and then we try to move these compounds through the process. And ideally, and this happens indeed in our flagship compounds, then partner them with either industry, out-licensing them, or find public-private partnerships to move things into the clinic. And this is what was also important in the development on site, to get much closer to the medical school, but also to interact much closer with what has been a major focus here on site is the informatics department. Some new members to the team. I'd like to welcome Florian, who recently joined, coming in from Jörn Piel's lab. You'll get to know Florian, talk to him. He's an expert in unusual pathways and natural products. Toby joined. He's actually not here yet, but he's on the move. He is here. He signed, and he's putting up his lab. I'll tell you about this in a minute. And Tanya, his wife, also joined us as an affiliate. So we're really happy with the two of them as sort of the team that now finalizes the process of the expansion of our institute. So this is our team. If we add our affiliates who are located at Saarland University and have a minor, a side job at the institute, the way we organize this is we have people who are in the faculty of informatics. So our bioinformatics people are mostly in the informatics faculty. Some of them are also in the medical faculty. Most of us in the natural products, natural sciences, forgive me, faculty, and then in medicine. This is how we set up the institute. And a number of people have mixed positions between the university and us. A number of people mostly have their job at HIPS, others mostly at the university. All that counts is that this team works together well. So obviously, with this expansion, we need some more room. We originally built the HIPS building, which you'll get to see later, because the barbecue will be right here. So this is the east entrance. If you walk down this street, east entrance, approximately 500 meters, you reach the HIPS building. This used to be the closest spot to Braunschweig. This is why we set it up there. It didn't make a big difference in the communication, but it's a good sign to be at the east entrance. And now in the expansion process, we'll have a first phase where a new building is just started to be set up. And this is where Toby's team will be housed. And we'll see another building even further to the east being set up, hopefully at a speed that we will all be happy with. So that's all I wanted to say here. This is the HIPS parking lot. We are up here, approximately where this building here is seen, and we'll do the barbecue down here. And here where you see the fence, this is where the next building will be set up. So we've seen some developments with the university, one that we call Pharma Science Hub, which is a closer interaction also with local and European, mostly industry, the medical faculty, and also the informatics departments. And these developments culminated in a Center of Excellence application called Next8, where Anna Hirsch and Martina Zesta and Andrea Volkamer serve as the speakers, which make it into the second round. And it basically has this topic of development of drugs, drug discovery in the conjunction with informatics developments. Let's call it this way. So for today, I look forward to having you here. Have a great program. We have invited lectures and talks. The sessions are as outlined here. We have session chairs who will introduce the speakers. So I think it's, again, a nice setup of very interesting talks to look forward to. We have traditionally a podium where young investigators from the HIPS will present their work. We currently, or we do have 360 registrants currently, so that's a record for this meeting. And we look forward to having this next year again as a two-day event, also to celebrate 15 years of HIPS. If you have questions after the talks, obviously the chair will guide you through. We also have poster sessions. Posters are surrounding you here. And we ask you to vote for poster prizes, which will be awarded at the end of the meeting. You can give them stars. You just can do this electronically. Here's the link again, but it's also in the menu. Please do so we can get some awardees who hopefully deserve their prizes. So with this, I'm already at the end. Thanks again for coming. It's great to see the HIPS community grow. I'd like to emphasize again, all of this would not have been possible without the team. So the HIPS team is what is most important to us, but then of course there's a lot of people surrounding the team. For us this means the HZI, Saarland University, the local government, who all helped us quite a lot in setting this up here. Now enjoy the day. I hope to see you during the day and also during the barbecue. Make use of all sorts of opportunities to exchange science, set up future collaborative efforts, and enjoy the day. Thank you so much. And I hand over to Andreas, who may not be known to everybody. He is the head of our bioinformatics department and will chair the first session.
Thanks. Wow, amazing to see that crowd. Good morning. So it's my pleasure to host that first session, and I will introduce immediately our first speaker. And while I'm introducing him, we maybe can put up the slides already. So our first speaker is Chris Cofield. He studied chemistry in Manchester before he moved to Oxford. And in between, there's of course many different things he did. And he's a truly distinguished researcher. He works on antimicrobial resistances. He works on the role of oxygenases in expression and protein biosynthesis. And many of these things, as I said, are truly amazing. He has papers in all big journals. I think there isn't any big journal where he didn't publish anything, right? And these papers are also cited hundreds, if not thousands of times. One of the things that impressed me most in his career, I have to say, he's fellow of the Royal Society. And another thing that impressed me quite much is if you read these abstracts of the manuscripts, you could get the impression he's just a basic researcher doing great basic research. But as you can already see from his title, it says from mechanisms to medicine. So there is a huge translational aspect also in his research. And that's what I'm excited to hear about especially. So now, Chris, stage is yours. And I hope this time the slides work. If not, you also have to tell us stories, because I can't tell stories as Rolf does.
Yeah, all fine. So thanks, Andreas, for the kind introduction and invitation to speak here today. So first thing I'd like to say is it's fantastic to see so many young people in the audience. And I'm going to give you a talk which is really about enzyme mechanisms. And I'm a trained chemist. I was always interested in biology and medicine, but perceived that I was rubbish at remembering things. And you have to remember less in mechanistic chemistry or maths than you do in medicine or biology. So studied chemistry. And I was particularly interested in enzyme mechanisms. I was inspired by someone you won't know called Bob Ramage, who was a peptide chemist before his time, who worked with Woodward on the total synthesis of cephalosporins. And he showed us the initial stage of the biosynthesis of penicillins. And I thought that was a fantastic reaction. And that's this reaction shown here, where a simple dipeptide is converted through to the penicillin nucleus in a single enzyme catalyzed step. And I came to Oxford to work with Jack Baldwin and Edward Abraham on this mechanism. Jack didn't have funding, actually, to work on the biosynthesis at the time, so he put me on a total synthesis project, which was good for me, actually, still the best training in chemistry, actually. And I'm going to tell you about some stories, and that's the story of enzyme mechanisms. Two diseases we're interested in infectious disease, antibiotics in particular, and cancer as well. But I'm just going to tell you some things that I'm interested in, which is the enzyme mechanism stuff today. Another thing to say is that chemists in the audience, we're trained to think in terms of functional groups. And when I started off, I used to sit where you are, and people would put up structures of proteins and things like that. And I'd always feel very inadequate because I was looking at the carbonyl groups and I was all overwhelmed by information on the slides there. So if I do that to you today, if you're a chemist, don't worry about it. That's my fault, not yours. And I'll give you a little bit of career advice as we go along as well. I think from my background, when we were growing up, there was a paucity of information, just having enough books to read, etc. We struggle to have that now. For the young people today, you're completely overwhelmed with information. It's just, selecting what to read and study is a difficult task. So again, don't worry about it too much is my advice. Become an expert in something. Take your time. You've got time on your side. Career-wise, I think you need to be reasonably good at something, and everyone in this audience is good enough. It's important you enjoy what you do as well, and I still enjoy what I started off doing. And finally, you need to get paid a decent salary so your parents are happy as well. I think that's all you really need in life, apart from friends and a social life. So I came to Oxford to work on the famous penicillins. And penicillins work, and I appreciate there's lots of, I'm skipping lots of details here in this talk as well, but we will do some details. They work by forming stable ester complexes with their penicillin binding protein or transpeptidase targets. And then a major mechanism of resistance involves beta-lactamases that learn to hydrolyze this ester complex here, so-called nucleophilic serine beta-lactamases. There's a second class of metallo-dependent beta-lactamases that catalyze hydrolysis via zinc ion intermediates. And for a long time, I thought those were very different. They have different structures, different evolutionary origins, but in some ways they're similar. So in essence, it looks more complicated than this, but these reactions involve addition of either oxygen atoms or an oxygen atom with two hydrogens attached to it, otherwise known as a water molecule. And I'm still interested in the details of these mechanisms. And stating obvious in a way, here you're going from a completely inactive dipeptide through to arguably the most important class of medicines, small molecule medicines that's been discovered. Vaccines still more important. And here you're going from an active compound to a completely inactive one. And one of the problems we have in a lot of our research is connecting biochemistry with physiology, and we usually fail. At least 19 times out of 20, we fail to do that. It's a long history of penicillin research in Oxford. Dorothy Hodgkin, in my opinion, the greatest Oxford chemist, solved the crystal structure of penicillins, and that revealed that the nitrogen of the beta-lactam ring is sp3 hybridized, so it's a tetrahedral geometry there, which mimics a high-energy conformation, one might propose, of the substrates for the transpeptidases. So there's more to the penicillins than the covalent reaction as well. They might argue they mimic the transition state of the reaction, and I'll touch on that later on. And when we solved the crystal structure of the penicillins, and it was on a Sunday, so we were working hard, solved by Ian Clifton, working with Jack Baldwin, and we did the usual sort of blast-type searches, wherever they were called then. And it was immediately apparent that related enzymes were very widely distributed throughout aerobic biology, not archaea for reasons I don't understand, including in humans and plants. So a large part of my career has been involved in assigning functions to these enzymes. And most of them use 2-oxyglutarate as a co-substrate and oxygen as well, and they catalyze the typical reaction, hydroxylation reaction, 2 electron oxidations there. And when we started, there were really only 2 categories of these enzymes that have been shown to catalyze these reactions. Pioneering work from the US and Japan had shown that hydroxylations occur of proline and lysine residues during collagen biosynthesis. And less famous work, but epidermal growth factor-like domain hydroxylases work from the US and Japan also undergo hydroxylations. So that work took us into all kinds of different areas we couldn't possibly have anticipated. So one thing is, if you do basic science long enough, then useful things will come out of it. So if you have a choice, in my opinion, start off with very basic questions, or start off trying to solve global warming or infectious disease, tuberculosis, that's fine. The danger is starting in the middle. So when you're young, if in doubt, my bias is to start with the basic science. So it's taken us into mechanisms mechanisms by which humans and other animals sense oxygen, epigenetics, how fruits ripen, and all kinds of other interesting biological areas we could never have anticipated. And so enzymes that are related to those that make penicillins are involved in all of these processes. And just to list some of them here, and don't worry about the details here, DNA damage repair, chromatin modifications, epigenetics, we put a lot of effort into that, modification of ribosomes, regulation of translation, and many other things there. But about, so that was me learning about biology and working in a highly collaborative way with many other groups there. But about 12 or 15 years ago, I decided consciously to go back to the antibacterial work, because the problem was there just wasn't enough people working in the field. And if you had a meeting like this, Rolf, 15 years or so ago, you've been lucky if you had 20 people in the room. Now you've got hundreds working on it. So it's fantastic to see that difference. And also, I came in at the tail end of the golden age of antibiotics in industry, and the best science was done in industry then, in my opinion, no question. But that's changed now in this field and other fields where there's unmet medical needs. Stroke and mental disorders would be two other fields where I think there just isn't enough work going on. So I'm going to, the first part of the talk is I'm going to tell you about mechanisms I'm interested in, and I don't know why some of this is useful or will be, but I'm going to tell you about it because I'm interested in it. So the 2-oxyglutarate-dependent oxygenases use 2-oxyglutarate as a co-substrate, and the substrate binds, sometimes dysplasia water molecule, enabling a vacant coordination site for oxygen to bind. And you have oxidative decarboxylation, don't worry about the details of that, to give an iron oxo species. And this does the reaction I was interested in as a synthetic chemist, the chemically challenging hydroxylation, which is via a sort of radical rebound type mechanism. What we're really interested in now, the fundamental stuff, is exactly how oxygen binds to the active site there. And it binds reversibly and then undergoes an irreversible reaction. And that's relevant to human oxygen sensing. So arguably, we've worked on the penicillin-forming reaction I've told you about, but hydroxylation, post-translational hydroxylation of proline residues in a transcription factor regulates levels of erythropoietin, and that in turn regulates levels of red blood cells. So I'm skipping lots of details here. And if you inhibit this enzyme, prolyl hydroxylases, that upregulates erythropoietin, and in turn upregulates red blood cells, and that is a new treatment for anemia. And based on small molecule work we've done, there are now, 4 or 5 approved drugs. We've recently, and only just published on this, published that the same mechanism, it might be useful, or it's useful at least in curing mice of leukemia. People's a completely different matter. If you regulate the transcription factor by inhibition of these prolyl hydroxylases, that's a useful treatment potentially for leukemia as well. This is a view, so I overwhelm you with data here, a view from a crystal structure of these enzymes. And the first thing I want to do is to illustrate how little we might know about these enzymes really. So the textbooks will say there's a single active site ion, which is coordinated by 3 protein residues, typically 2 histidines and a carboxylate, though sometimes it's a glutamate there as well. But in fact, in work on the epidermal growth factor-like domain hydroxylase, which catalyzes hydroxylation of asparagine and aspartate residues, crystallographic studies have revealed this enzyme just uses 2 histidines. This is a manganese substituting for iron for crystallographic regions. But the enzyme, which is mutations to which are linked through to disease, in which we don't understand at all, will work with just one single imidazole ligand there, 1 histidine. And even with just 1 carboxylate ligand, we can substitute it as well. So that was surprising, and you wouldn't have expected this from a chemical perspective. And one thing I have learned over many years is do not trust chemical intuition with respect to biology. Experiments are king. It's fine to have ideas and proposals, but don't trust your chemical intuition there. So these enzymes will work with just a single protein-based ligand, which means we might have massively underestimated the number of oxygenases there, because we do the bioinformatics searches based on particular motifs. So experiments are important. I'm now going to tell you about recent mechanistic studies on the penicillin-forming enzyme that we came back to. A key person in this is Patrick Raber here. Germany has recently started his independent group in Oxford there. And this work involves use of X-ray lasers, where you can generate a very powerful X-ray beam and collect a data set on a femtosecond timescale, which means in principle you can make movies of reactions. In practice, it's more complicated than that because you need to merge thousands of data sets to solve the structure, and I'm skipping an awful lot of details here. But we were interested into whether this technique could be applied to the penicillin-forming enzyme there. And our strategy was to make an enzyme substrate complex. So iron, this is the substrate for the penicillin-forming reaction. It's a dipeptide in which the sulfur coordinates to the iron under anaerobic conditions. And then select for a crystal form where the product will be trapped due to lattice interactions there. And that meant we could focus the mechanistic studies on the intermediates there. I appreciate substrate binding and product release are as important as the chemical bit of the reaction there, but you can't do everything. So Patrick has been working on this. During the course of this work, he's made over 10 grams of the protein to do these experiments. Now, you don't need that for every experiment, but another point is there's a huge amount of hard work that went into many of these slides that you don't see. So not 10 milligrams, 10 grams of protein went into these experiments. It's a collaboration between chemists, physicists, and biochemists, structural biologists, as we are in this thing. And in essence, the experiment involves a reaction chamber and microcrystals of the enzyme are sprayed onto a belt that goes into the reaction chamber where they're exposed to oxygen, then thrust out of the belt into the X-ray laser beam there. And you can also simultaneously measure the oxidation state of the metal by X-ray emission spectroscopy. And I'm not going to talk about that. I'm just going to show you the results at the end of the day. So if you do that, to illustrate just some of the people that have been involved in this work. This is our lab in Oxford, collaboration with Alan Orville at the X-ray Hub, but the physicists and engineers and data analysis are vital as well. So I'm just going to show you some pictures from this. So this is the starting anaerobic complex here. Reasonably high resolution there. If we expose that to oxygen, we can convert it through to the penicillin complex there. At relatively short time scales, you see electron density here, and I should point out, none of these are unique species. They're always blurred images to some extent. But the secret of this is to trap and you have to cheat as well. And I've already told you one way in which we cheat. So the first intermediate we can characterize is a dioxygen complex there that is likely at the superoxide oxidation state, and the X-ray emission spectroscopy correlates with that. So longer time scales, and this was unexpected and not predicted by the quantum mechanical molecular modeling studies, right, as well. The sulfur, and these have all been reproduced multiple times, the sulfur comes completely off the iron there. So more than 2.5 angstrom difference there. Here it's coordinated, it comes off the iron, and this, for reasons, I won't go into all the supporting evidence here, we're very confident is the thioaldehyde oxidation state there. So that wasn't predicted. Then reaction proceeds to give the monocyclic intermediates. This is the first time it's been observed under catalytic conditions. You might note we're cheating here by putting a deuterium on the valine, so slowing down the reaction of the second ring step there as well. So you can see that monocyclic intermediate there. There's been a rotation in the side chain of the valine residue there. So this CH that needs to be cleaved in the final enzyme catalyzed step is now projecting towards the iron, whereas in the initial complex, it's presenting away. And then finally, at extended time points, you see the penicillin complex there, beautiful electron density maps there. If you leave it for a very long time after that, you start to see the iron leaving the active site. And that's a crystal artifact. The penicillin remains rock solid there. So these are at least stills in a movie, and then you can start to use modeling studies to look at these, exactly how they're formed in more detail. Of more general interest, and again unexpected, was we started to see movements on the surface of the protein that again weren't predicted. Now there are some modeling studies on these and other enzymes that do predict conformational changes, but these particular ones weren't predicted. So if you look at this helix, for example, this is at the superoxide complex state. It's gone red, it goes cold again, heats up a little bit. These are crystallographic B factors here that give an indication of how much mobility is there, and then cools down again. There's also this bit on the surface here, gets hot, cools down a little bit. That remains hot, we think, because the penicillin is trying to escape from the active site and it can't move. So I think this is the thing that's more generally interesting, exactly how a beta-lactam ring is formed, a limited number of people involved. But what this technique does give is a global perspective on conformational changes during catalysis. Now, in principle, you can get that from other techniques such as fluorescence as well. In practice, it's difficult to do that. So are those motions relevant to catalysis? So I've just chosen one example of that. And again, experiments are important. So this is the iron here. There's a chain of 3 water molecules come away from the iron through a series of other residues, then there's links to the surface. So we've made multiple mutations to explore this. And I just want to tell you about one which gives an unexpected result. So this is normally an asparagine at this position here. And if you mutate it to an aspartate, in the crystals, the reaction is little bit slower than the wild type, but it still happens. You still very clearly get the penicillin products there. If we solve the crystal structure of the asparagine versus the aspartate, identical, of course, because you can't distinguish between nitrogen and oxygen at this resolution. If you do the biochemical studies, and even if you mutate the asparagine to alanine, you still get the penicillin, so no problem at all in solution in the crystals. So in the alanine, you get the penicillin. This is now solution data. Glutamine gives the penicillin, but in solution, the asparagine to aspartate mutation gives a completely different product. Doesn't give the penicillin that we see in crystals. You'd think this was a very conservative mutation there, but in fact, you get this intermediate here, this hydrated formyl glycine there, that leaks off at the monocyclic intermediate stage. And I'm not showing you the evidence for that, but the point is, I trust the experiments, not prediction as well. So why is this? So it might be this is a special reaction where conformational changes, where you go from the flat amides to the sp3 hybridized penicillin there, it requires special stuff to change the conformation there. So you're going along a sort of energy ridge there, and if you fall off that ridge, and we have other striking examples of this where you get completely different products. But again, you know, don't trust chemical intuition. It might be special, it might not be. You predict, say, catalase, for example, where you've got heme and hydrogen peroxide. You wouldn't need to do these conformational motions, but we don't know that. Just to summarize from this bit, this kind of global information is not readily obtained by any other method. Whether it's a good use of taxpayers' money to spend 2 billion, or more than 2 billion, whatever euros, pounds on these machines, is another question. The strategy starting from the substrate complex and ending with the product complex did genuinely reveal unexpected mechanistic insight. I should say, what you see in these talks as well is the experiments that work. You don't see all the ones that don't work. We've put equal effort into metallo-beta-lactamases with this method. Nothing useful has come out of it at all, for reasons we can discuss. Structural elements far from the active site are involved in catalysis, and I haven't shown you solution that is supported by solution studies. So I think from the medchem perspective as well, and I like mechanisms as well, I think industry is still pretty good at filling in holes of whatever it is, bromodomains. But in academia, I think we should think about new ways of doing medchem, whatever it is, PROTACs or whatever you like, and modulating enzyme activity, including increasing enzyme activity or allosteric type things. Mechanisms for that might be suggested by this type of analysis. Many mutations involved in antibiotic and cancer resistance are actually quite distant from the active site as well. And it's not obvious what they do from classical Michaelis-Menten type kinetics as well. So maybe this mechanistic insight will come from it. What this has also exposed to me, and I'll give you another example of this later on, is how little we really know about how small molecules, for example, oxygen and metal ions, move through the highly concentrated biological milieu inside cells. So it just finds me remarkable that things can diffuse within that super concentrated environment. Interested here in how getting oxygen to diffuse as rapidly as possible, because that's a limiting step in catalysis. But the fundamental topics like diffusion inside cells, I think, is of interest. So I now tell you a little bit about our work on beta-lactams. I have strong emotional attachment. A danger is that your PhD, you never escape your PhD work as well, so choose carefully what you do. The beta-lactams, any way you count it, remain the most important antibacterials of doses or life saved or money, whatever. And that's despite resistance. So certain types of chemistry, I would argue, are more useful than other types of chemistry. So thinking about what types of functional groups are most useful in terms of infectious disease is worthwhile. We're fortunate to given a donation to work on antibacterial stuff by a company called Ineos, now most famous for buying Manchester United, but they're petrochemical industry. And they gave a donation to my colleague Tim Walsh and myself to work on antibacterials. And one of the work streams, I'm not going to talk about that today, involves attempting to minimize use of antibacterials in farming, in particular meat farming. And I didn't know until a few years ago, the biggest use of antibacterials is in farming, more than 75%. And more than 75% of those used in farming are human drugs. So principally, there are others, ampicillin, but principally ampicillin, tetracycline, and colistin. And if you've heard this for the first time, you might think this is crazy, and it is crazy. But these are commodities, so they cost the same price as a cup of coffee I've just had here. So it's not that easy to replace that. I mean, the solution, of course, is to stop eating meat, but that's unlikely to happen in the short term. So this is a good field to work in. And generally, it's important that when you're young, you work in newer and fashionable fields. So it's okay, there's not many people working in your field when you're starting on. So I'm going to tell you a little bit about the metallo-beta-lactamases, and I've said that penosones work by forming a stable ester complex with transpeptidase enzymes, and the serine beta-lactamases involved from an ester intermediate there. Metallo-beta-lactamases have a broader spectrum activity, and for a long time were considered an academic curiosity. And we were part of a European network that was very successfully started to work on these enzymes, led by Jean-Marie Frère at Belgium, when there was no clinical application for the enzymes. But now that situation has changed. Tim Walsh is perhaps most famous for naming the New Delhi metallo-beta-lactamase, for which he was banned from India for a number of years. And a challenge in this field is that there are different subclasses of metallo-beta-lactamases. That's one challenge, so you've got to get a breadth of activity. The second challenge is, and this again followed from the work on the bacterial enzymes, there's 20 or so human metallo-beta-lactamase fold enzymes, which have crucial roles in human biology as well. So you don't want to inhibit those. And I'm not going to discuss this in detail, but we've done a lot of work with a colleague, Peter McHugh, on the SNM1 family of nucleases. And these are one of the very rare classes of nuclease that can eat past double-strand cross-links there. So mechanistically interesting as well. Remarkably, just as an aside, some beta-lactams inhibit the human nucleases as well. It's likely, though no one knows, that the true metallo-beta-lactamases evolved from nucleases. And as an aside, nucleases are remarkably understudied enzymes. To my knowledge, no known drugs target nucleases. Even from a mechanistic perspective, there's gaps there, despite their importance in both molecular biology and disease. So this is a more complicated mechanistic picture of the metallo-beta-lactamases, in which water is polarized to react with a cephalosporin, for example, to give a tetrahedral intermediate, then hydrolysis. And they have a different fold, and I thought different mechanisms to the serine beta-lactamases. So for a long time, thought it would not be possible to get inhibitors that inhibit both classes of enzyme. But one class that looks particularly promising, and this originated from work from Edward Abraham and then in European work, is boronate-based inhibitors. And again, I'm just going to show you a little bit, some pictures here, because they speak louder than words. So these bicyclic boronates, and we've worked on several different classes, potently inhibit both serine beta-lactamases by forming a stable tetrahedral complex there. They do inhibit the transpeptidase enzymes, penicillin binding proteins as well, but they're not particularly potent antibiotics as yet for reasons we don't understand, even though they inhibit them. Rather more surprisingly and pleasingly, they inhibit the metallo-beta-lactamases as well. Again, after the fact, it's obvious by forming a tetrahedral complex that bridges between the two zinc ions there that mimics the tetrahedral complex in beta-lactam hydrolysis as well. So if you, the secret was to ignore the protein structures, basically, and think about the chemical mechanism there. We like boron as well, as an aside, because then we've multiple examples of this, at least crystallographically. Sometimes unexpected things happen. So this is an example with structure solved by Phil Hinchcliffe in Jim Spencer's group here. The bicyclic boronate, the amide side chain of this bicyclic boronate, comes back in to react with the boron there to give a tricyclic ring system there that locks the complex at the active site at the enzyme as well. Again, we didn't predict that. So boron is nice because it's probably the wrong generation for these. It was a generation of power, a number of people, children, Power Rangers there, that boron can morph. So again, this comes back in some ways to Dorothy's beautiful crystal structure, the penicillins, where penicillin is sp3 hybridized here. Boron's interesting because it can mimic, in principle, both the sp2 hybridization state of the unreacted beta-lactam ring there and the sp3 hybridization state at the enzyme intermediate stage there as well. A lot of work still needs to be done to make those into really powerful medicines. In a second line of work, and I think this relates to what's going on at HIPS, because I was brought up when the pharmaceutical, I was in awe of the pharmaceutical industry, both in what they could do to bring things together and in terms of the power that they had. So the European lead factory was an initiative set up by the Innovative Medicines Initiative to do high throughput screens for academics. So we developed a screen for the metallo-beta-lactamase, and a number of compound series came out of that, but the one that interested me from a mechanistic perspective was these indole carboxylates. Again, crystallography was crucial. That trapped a water molecule at the active site there. So the crystal structure, we picked up on that, and collaborative efforts across multiple academic groups and different sites as well. So this collaboration can work to deliver useful things with support from industry. Led to the development of the indole carboxylates that inhibit, have very broad spectrum activity across different classes of metallo-beta-lactamases. And they're probably the most broad-spectrum metallo-beta-lactamase inhibitors that have been developed there. And the current focus is moving those on to phase 1 clinical trials. And I probably should finish there. Is that right? Thanks. I'm just going to skip to the end, though. That was one of... No, we don't have that much. I've been told to stop. It's all right. I just missed... What? I just skipped one story there. So yeah, I do think that point about collaboration is really important, and how to organize that across multiple sites as well. How to look after the people that are doing the work. I think the first author, last author publication thing doesn't work in highly collaborative work if you're trying to do translational work as well. So how to fix that and reward people from a career perspective, I think is important as well. And I'll finish off, despite the tragedy of Brexit, we are still a highly collaborative group as well, and welcome people from all over the world. And as I say, I've just given you highlights of the work today. There's a huge amount of work gone into this, and again, I think in particular to pick out Patrick's contribution for the work on IPNS. And I'll finish there, and happy to take any questions.
Wow, thank you for that amazing talk, Chris. The people that suffer working for me know that I'm a strict timekeeper and hate me for that, so never mind. We have time for 1 or 2 quick questions, but you know that that conference from the last years, we are really interactive, so we have the coffee breaks as well that you can then later on discuss with Chris. So the first question, Christina, right, first row.
Hi, thank you for this beautiful mechanistic work. I have one question to the oxygen, because that was kind of skipped. What is it a triplet state, singlet state? So what's the spin system of the oxygen and the iron? Does it matter?
Oh, yeah, absolutely. I mean, most of the oxygen around us is triplet state, otherwise we catch fire, I guess. I think early on in evolution, triplet state oxygen, whenever you've had transition metals and oxygen, you will, and water, will be some oxygen around. So yeah, it's triplet state, then it's activated by electron transfer from the ion to give a superoxide. Now, exactly where the electrons sit after that point, and with relation to the ion, and it's not just the ion, it's the ligands as well that matter. So it's not enough just to consider the metal. And as I indicated with the water story there as well, you're 3 waters away, and the exact protonation state of the water 3 molecules away from the ion is affecting the chemistry on the dioxygen-derived molecule.
Yeah, because most things were shown as radical reactions, if at all, in the mechanism. So I was wondering if that changes at one point.
Yeah, that's a good question as well. So certainly there's, in a number of these enzymes, there's evidence for radical intermediates. Principally, that's under biased conditions, where you either have a substrate analog or you have, you're using isotopes there as well. So it's actually quite tricky to prove radical intermediates in the wild-type reaction. My suspicion is the wild-type reactions, or the natural reactions, are more concerted than is sometimes proposed.
So let's have a second question, if there is one. If not, I think it's...
Rolf is good.
Oh, Rolf. How could I overlook Rolf?
Yeah, thanks, Chris. You mentioned you nailed the thioaldehyde, right? I was wondering how, using this technology, you could nail the thioaldehyde.
Okay, so I didn't mention that for young people. Sulfur biochemistry inside cells, really good topic to look at. But it's technically difficult, so it needs somebody in the lab doing it. It's not somebody like me supervising someone. You need to be in the lab, because as soon as you lyse the cells, everything starts shifting. So even disulfide patterns inside cells, there's no good way of measuring that. So I didn't go through all of that. So the thioaldehyde, if it leaks off, it tautomerizes to the enethyl. And then enethyls do all kinds of interesting dimerization reactions, including making mixed disulfides rather than homeric disulfide. So you can reduce the thioaldehyde by another thiol reacting at the sulfur, or the sulfur of the thioaldehyde to give a mixed disulfide. Or it can react on the carbon of the thioaldehyde to give a dithiolane type structure. So it's complicated. But good. Interesting.
Good, perfect. So let's thank again Chris for finding your way here to Saarbrücken.
Thank you, Andres.
Amazing talk. And you will recognize that our session is a journey through Europe from the north to the south. So our next speaker is from Denmark. It's Anja Boysen, and she studied physics in Roskilde. Thereafter, she did an industrial PhD and later became professor at the DTU in Denmark. And her work is on nano and micro mechanical sensors, but also on the oral application of drugs using microcontainers. Also, if you go over her CV, it's really spiked with many different things, and I again picked some highlights that I thought are perfectly interesting and amazing. So first, she has an amazing track record. She has an age factor approaching the 80s, which is wonderful, and manuscripts cited hundreds and hundreds of times. But it's not also the publishing track record that is amazing. Also in attracting funds, she is really recognized. And for me, one of the most important things was her the ERC grant. So also translational aspects in your research, and today she will tell us something about microcontainers that we can use for anti-infective or infection research. My pleasure having you here. Welcome to stage.
So thank you very much for the introduction, and a special thanks to Klaus Michael Lehr for suggesting me to be here. It's really a pleasure. So yes, my background is actually, I'm trained in physics and in math, and then as you can see, I've moved a bit away from that field, so that's maybe also a message. You can start in one field and move a little bit across fields. So before jumping into 3 stories that I have for you, I have one on sensing, I have one on oral drug delivery, and then I have one on in vitro systems. I'll just spend 2 minutes on introducing who we are. So we are a center of excellence that we call Idun. Idun is one of the Nordic goddesses. And we kind of have a motto, bringing science to life, that I'll come back to. But you'll see that we like to dare better science. We like to grow better scientists and also to create new businesses. So I'll also show you a few examples of how we translate into startups. So the Center of Excellence that we have now, it's been going on for 9 years. And it started by us getting 2 major grants at the same time. You never know when this happens, but it just happened. So we got one to continue what we've been doing for many years in sensors and micro and nanomechanical sensors. And then we got a new grant on using micro and nanostructure for oral drug delivery. And since I was not allowed to coordinate 2 major projects, they actually asked me to merge them into one, because then I was only coordinating one project. And I would have to say that what started as a very forced marriage is now a really, really happy marriage. Because, you know, honestly, a lot of the sensors that we had been developing, they didn't have any real applications in mind. But once you start collaborating with pharmaceutical scientists, medical scientists, you get a lot of new inspiration. And there's a lot of cross-inspiration in between the fields. Later on, we got other grants to support activities, and I'll share some of those stories with you as I go along. Is the sound okay? It's a little rattling, okay. But in terms of bringing science to life, it's our ambition to apply our top-down micro and nano technologies for life science applications, but it also kind of covers that we really like to translate some of the things we do into, for example, new businesses. So that's why I've tried to mix in some of these stories as well. These are some of the companies that we have been spinning out. And there are 3 more in the making. So you'll hear about some of these stories as well. So this is us when we're gathered. Maybe you can see that we are from many different countries. We're from 25 different countries. So it brings in a lot of diversity. We're also from many different scientific disciplines, from computer science to pharmaceutical sciences, medical sciences. And we're also collaborating with University of Copenhagen and SSI in Copenhagen. If you're interested, you can follow us on LinkedIn. So I have 2 main stories, one on therapeutic drug monitoring. It started actually as therapeutic drug monitoring in cancer, and then it actually moved into infections. And the story is on oral drug delivery. So the first story is the therapeutic drug monitoring. And this started when we were approached by the head of the oncology unit, pediatric unit in Copenhagen, because when they're treating children with leukemia, they have to give them a very high dose of a drug called MTX. And they need to monitor these children very, very closely. Because this is a very toxic drug, they get it in a high dose. And since these children are very sick, they metabolize these drugs very differently. So they need to take blood samples every second hour and measure. So you need to stay within the therapeutic window. Overdose, you might even kill the patient. Underdose, have no effect. In Denmark, the central lab, they can do these analyses every second hour, but they would like to do it more or less line next to the patient. And in other hospitals in Denmark and in all over Europe, you cannot do this treatment because you don't have the facilities to monitor these patients so closely. So then we started actually looking into how can we do this, and we got a new grant from the Novo Nordisk Foundation. I've never done anything like this. So the deal is you get money for 3 years to hire researchers to go into a project, and after 3 years, you kind of promise that you're going to start a company. So I started hiring people in for doing the research, and then middle way through, we had to kind of face who wants to be in the company and who have other ideas. But that's another story I can share with you at another time. So these are the people that were hired in on the team, postdocs, to do the research. But you can see, which is kind of unusual for me, is that we also hired in business developers. So they, of course, had an opinion on where to go for the future. But first, the core team, again, from 6 different locations all over the world, and also with 6 different scientific backgrounds to actually make this work. What technologies we used were actually that we've for many years been working with something called surface-enhanced Raman scattering. So this is a paper over here, where we kind of did the first results on nanostructured surfaces for that. And then we actually also, before we started this journey, knew how to put some of these sensors into microfluidic platforms. And I'll show you an example of that. But just a few words about Raman. Maybe some of you are already working with Raman microscopy. It's a really nice technology. You shine a laser onto a surface. Some of the light might be inelastically scattered in the molecules you have, and the light comes back with a shift in wavelength. And it's this shift in color or wavelength that you kind of use to get a chemical fingerprint of whatever you have on the surface. It's label-free, and it gives you an entire spectrum. So you can really kind of know what you have, also in a solution. The drawback is that it's not very sensitive, so you can enhance the Raman effect by placing molecules on rough metallized surfaces. And that's what we have been doing for around 10 years now. So we actually started by being very interested in the micro and nanofabrication. So we etched silicon nanopillars, and we coated them with gold or silver. So this is Michael's initial work. And then they look like this. So these are nanometer structured nanopillars, and you have them on a 4, on a 6, 8-inch wafer. The cool thing is we didn't really understand why they were, because they worked really fantastically. They enhanced the Raman signal a lot, like 1,000,000 times or more. We didn't understand what was really going on. But then Michael, he discovered if you put liquid on these substrates, these nanopillars, and the liquid or whatever analyte you have evaporates, the capillary forces are going to bring your nearest neighbors together, and they form small domes where the metallized heads here, they touch each other. And this is the ideal situation for enhancing Raman signal. So imagine you had your molecules. It could be the drugs you're looking for. And then afterwards, you make these small metallized nanopillars lean towards each other. They're almost touching. And here you have what's called an electromagnetic hotspot. And that's really what enhancing your Raman signal. And you get this kind of for free. It's just happening automatically. Otherwise, people are really struggling to kind of make small nanobeats meet each other. So this, they actually, indeed, you can look at them afterwards in a scanning electron microscope, and yes, you form these small domes on your surface. You can cut these silicon pillars into small squares, and you can give them to your collaborators. You can store them in the lab until future use, and that's kind of one of our first examples of a little spin-out, where you can buy your own substrates if you like. So they, produce and sell to researchers who would like to develop their own protocols. Now going into therapeutic drug monitoring, we need to measure on blood samples. So before doing anything very advanced, Yarman, he just took and glued some of these small nanopillars onto a holder. Down here you have a little reservoir, aquarium. And then you just take your blood serum and your nanopillars, and you put methanol into the serum, and that's both to precipitate proteins in the blood serum and also to wet your SIRS substrate a bit more. And then you put it into your sample reservoir, and you dip the SIRS chips half the way into your blood serum. What's happening is that these nanopillars, they kind of work as a filter paper. So they're going to suck up your blood serum, and only the small molecules are allowed to filter through, and they're going to travel up into what looks as a dry region. And then you measure your Raman signal in the dry region on top of the SIRS substrate. And by doing so, it actually works kind of as a filter paper. All big molecules that you might still have left in serum, they are trapped. They're not traveling up. And the other ones, they'll stick and give you a really clean Raman signal in what looks like the dry region of this structure. So when we had those first results, which was just manually dipping something into a solution, we got the first blood samples from the hospital. So children not treated yet, and then treated after treatment, 24 hours and 48 hours after the treatment. We used the untreated blood samples to build calibration curves, and then we could try to see what concentrations do we get. And for one patient, you can see we got very much close to the ELISA assay run at the hospital, and for the second patient, we were 50% off. At that time, we knew, there were two main issues. One was the way we treated the data, and the other one is that everything is manual dipping something into a solution. You can imagine that many things can go wrong. So we had to solve both. First of all, we do machine learning. We use now the entire Raman spectra. So this improves a lot. We can distinguish not only MTX from other drugs, but also MTX from its metabolites. The second thing is moving away from a manual treatment, dipping, and putting everything into a fluidic platform. So we take these small SIRS chips and we put them on a CD disk. So imagine you had a normal CD disk, but now you engrave if you know what a CD disc is, by the way. At least you have heard of it, maybe. So it's a polymer disc, and then you engrave channels, and then you start centrifuging it. By doing so, if you place your blood sample in the center of the disc and you start rotating, the centrifugal forces are going to move your blood sample upwards. And by controlling the spinning acceleration and speed, you can do a lot of different protocols on a disc. You can do filtration, valving, pumping. You don't need any external pumps. It's a very simple system, and it never has bubbles inside. So we use this now to do all the blood sample treatment and also to dip only half of the chip into the blood serum. So Laura was the first one to integrate things here. First of all, we just integrated how to mix methanol with your blood sample. And later on, we also integrated pre-treatment of the blood samples. We have built several prototypes of the device itself that needs to go to the bedside of the patient. Here were the first two prototypes, and Roman is the one doing both the prototype and all the programming that goes with it. And then now, the startup. You can see now also the slides they've changed. They're much prettier than mine because this is part of the pitch deck for the company that's called Fluilux Diagnostics. So the business case then changes. That's also interesting for me. MTX is a very cool story. These patients, they really need to be monitored closely. But when you talk to the business developers, they're going to say, this is a very small market. Fortunately, very few children around the world, they suffer from leukemia. It's actually a problem for us to get enough blood samples. So as a first market idea, they have narrowed it down that we should focus on sepsis and the treatment of patients in the intensive care unit. In the intensive care unit, around I think 26% of the patients are overdosed because you don't really know how they metabolize the antibiotics. And you have another 30% of the patients that are underdosed. Overdosing might kill these patients. They already have organ failures. So there's a huge need for monitoring these patients very closely, and it has to be very fast. So that really makes sense that you have a bedside instrument for doing this. Still, when they go and pitch this work, they show the latest results related to MTX, because this is where we have been the most active. And you can see now we're very, very close to the HPLC standard and also to the immunoassay from the hospital when we kind of compare with the source data from our discs. So this is in progress. The company has not incorporated because we just got a European Innovation Council transition grant. So that's going to give additional 2.5 euros for this team to continue with soft funding. So I think in a year's time, they will incorporate. And here you find their contact info if you're interested. So that was my first story. I move on to my second one, which is related to oral drug delivery. And this is a story that started around 9 years ago. And we really kind of to make a contribution to move away from all the needle injections that are needed for all the drugs that we cannot take orally. You need to protect the drugs as they travel through your stomach into the gut, and you also need to pave a way to actually get through and into the bloodstream. We didn't want to make any new formulations. That's not our background. We wanted to make new devices. And we started maybe a little naively to think, okay, we'll make small microcontainers, like 300 microns in diameter. They are made in polymers. We're going to load them with drugs, coat them with polymeric lids. They're going to orient themselves in your intestine. And when they are in the right location, the lid will dissolve and it will release the drug. At least then you have done a good part of the way into your system. They look like this. They are around the same size as small sugar grains. And since then, we have moved on to a lot of different designs, depending on the applications and also a lot of the challenges that we have met during the years. You can load drugs in various ways. So this is just an example where these small devices are filled with powder drug. That's an easy way to do it. And you can coat them with pH-sensitive coatings like the ones you can buy for normally you would use them for coating bigger capsules. Before moving into the real applications, sneaked in another startup story, which is that I wanted to see where do we have the drugs inside these polymer capsules or small microcontainers. And then Oleksiy and Roman and Chiara, they worked on a home-built system where we can do Raman in 3 dimensions. So there we were actually able to map the entire microcontainer, and all the red part over to the left, this is where we have the drug. But then, of course, Alexei and Roman, they started also to play with polarized light. And by doing so, they could actually, in a tablet, not only do the normal Raman imaging, which is what you see to the left, where you would normally just be able to see where do I have drug and where do I have polymer, but they could also now see crystal orientation on top of a tablet. And this is normally something you would need a synchrotron to be able to do. So based on those Raman results, they started a little company called Light Novo. And besides big systems that can do this type of thing, the crystal orientation, they also do small micro Raman systems and some medium-sized Raman systems. And this one the very small Raman system just recently got published. So they're moving on in a, I would say, very promising direction. So small microcontainers. Before giving them to animals, or humans eventually, we put the small containers into larger capsules. And then we have been addressing a lot of different applications. We have been using devices for probiotics delivery, antibiotics delivery, insulin, vaccine, and also for a bit of gut sampling that I will not be able to talk about today. In some cases, it works fantastically. And I'll just show you an example of the antibiotics. In other cases, we failed horribly. So the first thing we wanted to see was, can we actually help eradicating bacterial biofilms, and can we do it better than just solubilizing antibiotics? So we loaded antibiotics into our small devices, and we collaborated with Søren Molin and his group at DTU. Maybe you have heard of them. They're very good in bacterial biofilms. And what we could see was that if we loaded small microcontainers with antibiotics and we used mucoadhesive coating as the lid, we could actually eradicate the same amount of bacteria and inhibit any bacterial growth much better than if you just had the same amount of antibiotics in solution. Actually, you would need 3 times more antibiotics if you just did it in solution. And this has to do with the way these containers, they embed inside the mucus layer. But then there's one story where you kind of always write in your, at least I often do, that we also want to do something with insulin because we have Novo in our bag. Novo, our insulin, is very difficult, as you know, I guess, that it doesn't want to go through the cell layer. And one case is that you can try to work with penetration enhancers, so small chemicals that will actually open up the tight junctions between your cells in the intestine. And then the insulin can go through. So we thought, okay, just going to mix permeation enhancer and insulin into our small microcontainers, and then maybe it will work. We'll try it out. It didn't work. We tried a lot of in vitro systems first, and we also tried in animals. And what we learned is that for insulin co-delivered with a permeation enhancer, you need to be kind of smack onto the intestinal wall. Otherwise, it's not going to work. We tried to, change the spacing a little bit, and the message was clear. I mean, you just need to be really close to the epithelium. So Lasse and Mehdi and Carmen, they came up here with a new idea to use a foil. So this now is, you could think of it as a lot of small cavities in an elastomer. So it's a foil you can roll up and you can put it into your big capsule. In all the cavities you have the drug, and it's coated again with a layer that's pH sensitive. And as soon as you release the foil in your intestine, when the big capsule dissolves, the foil is going to unfold. It really wants to regain its original shape. So that's what you can see here. It looks a little bit uncomfortable when it's just a piece of intestine and the foil has been allowed to unfold. But you can really see it's pressing against the epithelium, and now you are as close as you can get to the intestinal wall, I would say. So this is what you see here to the right. So in collaboration with University of Copenhagen, we did the first proof of concept studies back in 2020, and we could get a tiny bit of insulin, less than 0.01% across and into a rat, which compared to nothing was still kind of an achievement to us. Then we have improved. There were certain things that were not really working in the first case. So among others, the way we coat the foil initially. And now we have 2 to 3% bioavailability of the insulin just using the foil itself. and this is in rats. And then we also have moved to big pigs with the help of Aarhus University. And here we are working with an antibacterial peptide called Nisin. And here actually we can improve the oral the bioavailability by more than 300%. A few stories about getting even closer to the epithelium or maybe inside it. One case here is using micromotors. I don't know if you're familiar with them. But these are small devices that can swim by themselves. And in this case, we use magnesium particles that will, maybe you have tried to play with it in high school. So if you put it into a solution, you're going to create bubbles. So imagine a 20 micron magnesium particle that you coat with drug and a pH-sensitive coating, but just in the bottom there's a little hole so the magnesium can react. If you then put the magnesium into one of your microcontainers and you coat it, and then at some point the magnesium particles can get out, they're gonna swim by themselves out of the microcontainer and further through the mucus into the epithelium. And this is what we see. We can actually study how these micromotors, they come out of the microcontainer and swim through mucus. And they actually swim more in a straight line in mucus compared to just in a buffer solution because of the viscosity of the mucus layer. We also looked into how we improve the adhesion of our devices. So we made some arms that can attach to mucus. And here, again, we repurpose technologies that are already out there, not the DVD disc itself, but the blue laser from the Blu-ray player. You can take the Blu-ray player apart and just structure devices with the blue laser cross-linking polymers. And so this is a home-built 3D printer that can actually make devices that are very small, so we can do all the tiny structures on our containers, for example, that we need. So here we can do all sorts of containers. I can see my chairman is standing up. So this is also a startup team that is printing a lot of things for us using just the blue laser from a Blu-ray player. And besides our devices, they do a lot of microneedles, hollow microneedles. They do Eiffel Towers, which is just for fun, of course. And they're very proud of this cathedral with a lot of overhanging structures. So if you're interested, you can follow their work. They're happy to collaborate. So these were the two main stories. I just wanted to show that we have an ongoing project called Cell Loop, where we actually culture cells, human cells and bacteria, the disc as well. We are really liking these discs with channels. So just, you can see, you can culture cells in this little chamber. You have a supply of growth medium here, and when you have fed to the cells here, there's a waste chamber around here. Again, you are pressing the liquid through your chamber of growth by simply just spinning the disc slowly in an incubator. You're having a very stable flow, there's no pumping involved, and you can do a lot of very simple experiments in this way. And you can then also see that if you are impatient or you want high throughput, you can just stack a lot of discs on top of each other, and then you have a little motor that's spinning them all around, and then you're growing your cells inside. This actually started, this idea started when we were doing the collaboration with Søren Molin on the antibacterial biofilms, because this is what it looks in their lab. Look at all the microfluidic chips, all the tubings, the amount of buffer and growth medium they need, bubble traps and so on. All of this is gone now. It's just a spinning motor and a disc. So this is how it all got started, again with infectious diseases, I would say. So this brings me to the end. I would like to thank our sponsors and also you for your attention. And foremost, you know, this amazing group of people who make it so much fun to go to work every day. Thanks.
Wow, second mind-blowing talk. I start with a question. I was really fascinated how good the technical sensitivity of your blood assay is, how good the limit of detection is, that you're really approaching the same quality as an ELISA. Does it also hold for the coefficient of variation? So if you repeat that measures, it stays like that?
So the relative standard deviation is around 10% now, and that's where we should be. One thing that we have a lot of focus on is actually the SIRS substrate itself. I don't know if you're familiar with SIRS. Sometimes the substrate, it enhances differently. So we really need to make sure the way it's fabricated in the clean room and the way you store it. If you're not kind of wrapping it immediately in a protective environment, it's going to attract carbon and other things, and it's messing up your signal a bit. But you can do a lot with machine learning as well.
It's mind-blowing, amazing. So congratulations to that. Other questions from the audience here?
Interesting talk. I have a question regarding the microcontainers.
Yes.
So you were mentioning that you are also thinking about using them for probiotics, like putting bacteria inside.
Yes.
Do you have any data on this yet?
Yes, we have also some patients on that. I think actually the thing that works was when we were doing not microcontainers, but you will also find papers where we are doing layers of electrospun fibers, where we encapsulate the bacteria in one of the layers. And then you can actually, there's a recent paper also where we're showing, when you have these layers kind of protecting the bacteria, you can cut them in 3 dimensions afterwards with a tool, a bit like a cookie cutter, but it's also 3 dimensions, and then you just put in, and then you have, small particles in this way. Klaus.
Yeah, Anja, really, really great talk, and compliments about your achievements. I mean, especially also would like to talk a little bit more about the microcontainers. I think I've been pursuing or following what you have been doing since you about started with that concept, and impressive how you translated and developed it further. You showed there is a potential regarding penetration of mucus, regarding penetration of biofilms. Both are similar, but also dissimilar hydrogels. And I do not disbelieve at all that you can, let's say, increase the penetration through the biofilm gels or through the mucus gels. The challenge I would see is how do you make these particles that are moving by the little membranes towards the mucus, and because you have to overcome the liquid gel interface.
Yes.
And most systems would probably just gear away. So by the moment you have them intermix within the mucus or within biofilms, it will probably work without the directionality. But how do you see the problem that you give a direction towards the biofilms and not away from it, not at random, and to penetrate this interface where you're probably encountering elasticity models of a couple of dimensions? And yeah. So what do you say about that?
I mean, as you've seen also, we started maybe, I would say, a little naively with just containers and thinking they would, at least 50-50 would orient and they would sit somewhere in the mucus, for example, or in a biofilm. And it works for some cases. I think that we are actually moving more and more into some more brutal things. We're getting more mechanical. We're actually grabbing, somehow pushing things. The foil also, so we have tried a lot with different type of coatings. It all works really nicely in vitro. In vivo, no.
Maybe I should have asked more precisely the next step. I could imagine that you want to give a direction, kind of not just a non-specific movement in all directions, but
the motors,
giving
for example,
a sensing system that is you where to go and also move it towards a certain target.
Yes. We are working on that, and I think that's a really good idea to, for example, use some biomarkers that you can move towards a region. The micromotors, as an example, is something that has certain directionality. You can see that if you confine them, they can only go out. They can only leave the system in one direction, and they're already very close to the mucus layer. So they are coming out. And now we're working on micromotors that have more fuel, and they also look as small rockets. So let's see how that's going to go.
Oh, yeah. Thanks for a great talk, and I think a really good field to work in as well.
That's fine.
I had one technical question and a comment, if that's okay.
Yes.
Can you comment on the selectivity of the Raman method? Can you distinguish between drugs and drug metabolites?
Yes,
for
we
example?
can. Yes, we can. And that's actually kind of, we didn't know that from the beginning we would be able to do that, but we can actually clearly do that. And the ELISA assay cannot do that.
Correct.
So we actually have a selling point compared to when they run the ELISA, which is normally what they do in the big robots. HPLC is only for very special cases. It's too labor intensive and expensive.
Yeah, I mean, of course, the dream is to have sensing of the drug and delivery of the drug in the same device on the patient, as can be done for diabetes.
I mean,
now.
that's kind of what, I didn't cover that at all. We have a grant now, a Novo Challenge grant, which is on energy harvesting inside the gut. And the vision for us is that if we manage to do that, instead of swallowing a battery, we'll be able to combine some of our sensing activities with our delivery activities.
But I was a bit surprised, the business development people saying go away from cancer, because I think for chemotherapeutic delivery,
yes,
continued release would be a really interesting thing to study in diseases like multiple myeloma, for example, where you have multiple treatments. And the other one was suggested is antipsychotic medicines, because you're probably aware patients are often given high doses of injections, so-called depot injections, which is not really ethical, but because they won't take the dose. So controlled delivery for antipsychotics, maybe you won't make lots of money, but would be a very useful thing to do.
Yes, I agree completely. And I just have to share that sometimes it's actually a bit frustrating to work with the business developers once in a while, but of course they have a point. And we also got a grant from the Childhood Cancer Foundation in Denmark, so we are continuing the work on leukemia as a research project.
And in relation to cancer, of course, if you can get drugs into solid tumors, which are kind of biofilm protected, that would be a good thing.
That would be a very nice topic. Thanks a lot. Yeah.
I see our first two speakers will have sufficient time to discuss this over the coffee break later. So first of all, let's please thank Anja again for making her way to Saarbrücken. Before we continue our journey through Europe, I have to remind you that especially online folks can vote for each poster in between 1, 2, or 3 stars. So you can really distribute the stars to these posters. Now we continue our journey southwards. We arrive from UK, Denmark, now Germany, to our friends in Braunschweig that Rolf mentioned already this morning. And you know, it has a tradition for us not only to have the superstars here already as we had this morning, but that we also want to have the coming stars presenting here. And this is one of these talks. So we have Julia Würmann from the HZI in Braunschweig. She's from Mark Bernstrup's group, also in the same topic working as us. And she wants to tell us something about the quantitative analysis of the uptake of anti-infectives. So Julia, enjoy your talk.
Thank you very much. Hello and welcome to the third talk with the title Quantitative Analysis of the Uptake of Antibiotics in Pathogenic Bacteria. And as mentioned, I'm a second-year PhD student at the Helmholtz Center for Infection Research, and our group is particularly interested in measuring uptake in gram-negative bacteria. So first you may ask yourself, why is uptake even important? And uptake and accumulation is often the bottleneck in drug discovery, because antibacterial activity is determined by the potency, that means target inhibition, as well as accumulation, which is the net result of influx and efflux. And uptake is especially problematic in gram-negative bacteria since they possess an additional outer membrane. There are different methods to measure uptake, but we use the current gold standard, which is the quantification via HPLC-MS. In the following, I shortly want to introduce you to the 2 research aims in my thesis. And the first one is the new take on uptake under MIC conditions. And here we try to correlate antibiotic activity given as the MIC, minimum inhibitory concentration, with accumulation in gram-negative bacteria. And herefrom we learned that we need some further information, especially on the target and the molecules. And now we focus more on the group of fluoroquinolones. And we want to determine here the so-called target occupancy of these fluoroquinolones at their target, the gyrase subunit A in E. coli. That means we want to quantify the on-target site concentration that is required to inhibit the growth of bacteria. As you hopefully can see here, the fluoroquinolones bind the gyrase subunit A, and one molecule is binding one subunit. And one gyrase is composed of two subunits, so we have a defined ratio. And on the right-hand side, you can see the fluoroquinolones we're interested in. These are besifloxacin, ciprofloxacin, levofloxacin, and moxifloxacin. So how are we planning to measure the on-target site concentration? Well, first, the bacteria are incubated with the respective antibiotics, and then we try to combine 3 different assays, which is the cell counting assay, proteomics, and HPLC-MS. HPLC-MS, as I've mentioned, is used to determine the intracellular concentration of the antibiotic, and proteomics is used to calculate the DRA concentrations afterwards, so that we can set both into relation. The cell counting assay we think is important because the morphology may change after antibiotic stress, so after antibiotic treatment, and as an output, we get the cell count and the cell size. In the following, I give you a short overview on the procedure. So as I've mentioned, we have different concentrations, and after inoculation and incubation, we obtain these samples. And we then proceed with different assays. This is first of all the whole cell assay used for HPLC, MS, and proteomics, and cell counting. So let's start with cell counting. Here we use a specific dye and an enhancer and the loading buffer. And these slides are then centrifuged, and afterwards the quantum TX microbial cell counter is able to take 10 pictures and give cell size and cell count as an output. On the right-hand side, you can see the whole cell assay, which is basically cell harvesting and washing and sonification. And afterwards, these samples are subjected to LC-MS analysis, and here you can see one example of moxifloxacin. Now, we want to try to calculate the DRA concentration, and we want to do this based on one peptide. So first, you have to find the optimal tryptic peptide. So we used the bacterial lysate, which was digested and then subjected to LC-MS. And then we were able to find the peptides here highlighted in gray. And we then proceeded with one peptide, which we identified as the optimal peptide, here highlighted in red. By the way, this is not belonging to this part. These are 2 different peptides. As I've said, we decided to proceed with this one. And what we need now is the linear peptide synthesized and the heavy labeled peptide. The heavy labeled peptide is used as an internal standard, and the linear peptide is used as a normal calibration curve. As a heavy label, we choose the leucine, and you can see, maybe not, it's too small, but the mass spectrum, there's a mass shift as expected. Then we proceed with our samples. We reduce them, alkylate them, and digest them, and so that this wanted peptide here is generated. At the moment, we finished this step, and we now try to find the best conditions for chromatography for the aqua peptide, because the aqua peptide is then spiked to our samples so that we can set the aqua peptide and our normal peptide into relation. So I can only provide you with the first results, which is, for example, cell size. You can see that there's the tendency of an increase with antibiotic concentration, here depicted as the X-fold MIC, whilst the cell concentration, despite normalization, by the way, is decreasing, or shows the tendency to decrease with a higher antibiotic concentration. And on the right-hand side, you can see that we were able to calculate the molecules per cell, and maybe you can detect that for basifloxacin in red and for levofloxacin, we see a similar pattern with a lag phase, I would say, an increase and afterwards, plateau, while ciprofloxacin in orange and moxifloxacin in blue show also an almost continuous increase. As an outlook, we think that this assay could be extended to escape pathogens, so we tried the uptake assay with the gram-negative escape pathogens. For example, another E. coli strain, Klebsiella pneumoniae, Acinetobacter baumannii, and Pseudomonas aeruginosa. Of course, one has to consider that the gyrase may have a different amino acid sequence. Further possibilities are that we gain more information about cellular stress responses, for example, the regulation of protein expression, tolerance or resistance mechanisms. And we think that the combination of these assays may also be applicable to further antibiotics where a defined ratio is given. For example, trimethoprim and dihydrofolate reductase. Let's sum up. We tried to link antibiotic activity with uptake, but we realized that this requires more information on the target and the molecule. And until now, we were able to obtain more information about the concentration-dependent uptake of fluoroquinolones in E. coli. And we now want to determine the target occupancy by combining LC-MS, cell counting, and aqua. And we hope that this contributes to a better understanding of uptake and the actually active concentration of antibiotics that is required to inhibit the growth of bacteria. And with that, I'm at the end. I want to thank the Hosi Bio Group. Some of us are here today. Of course, my supervisor, the uptake team, and the CBRO group that did the proteomics measurements for us. Thank you very much. And if you have questions...
Yeah, thank you, Julia, for that talk. I'm sure we have some questions. I mean, the issue is, I'm a bioinformatician. I could not even ask a question on the topic myself, so I need you. Chris?
Oh, thank you. Yeah, very nice talk. So just one suggestion. It is difficult to measure the fractional occupancy inside the cell.
Yeah.
And one thing you might consider, and I appreciate there's be a lot of work in this, if you had an inhibitor that competed with your molecules, that reacted irreversibly and covalently with one of your peptides, then with appropriate controls, you can use that competition to give an approximation of the fractional occupancy. And we did that in a cancer field, and then GSK did it properly, if you like, afterwards. But that might just work with gyrase inhibitors, because you could put a photo cross-linking group on, for example.
Okay, thank you very much.
Perfect. So I have two more things ahead of the coffee break. The first one is the good news. The coffee break will stay for half an hour, and we start a quarter past and not clock and sharp. So you have sufficient time to discuss. That's the wonderful news. And then for most of the audience, we are done. I want to have the speakers coming up again, that we can take some photos together. And I want to take then the opportunity to thank all of you again for these wonderful presentations. It was quite fun for me. It was amazing talks. Thanks. All of you giving these presentations. And now enjoy the coffee break. Bleiben. Fotos. Ich hole die anderen hoch.
Hello, everyone. Please take a seat again. We would like to continue to stay on time. My name is Anna Hirsch. I'm the head of the Department for Drug Design and Optimization at the HIPS. And it's a real pleasure for me to chair the next session, which is dedicated to the young investigators at the HIPS. It's all PhD students from different departments. So our first speaker is Aljoscha Gabelmann, who is doing his PhD in the group of Klaus-Michael Lehr. Aljoscha, the floor is yours.
Yeah, hi together. So today I'd like to talk a little bit about my PhD project, the development of an advanced vaccine platform for mucosal immunity by the co-delivery of 2 mRNAs encoding for an antigen and an endogenous adjuvant. To make sure that we are all on the same page, I'd like to start with some background on mucosal vaccination. What are its advantages? What do we need to overcome? And our strategy to do so. So a major advantage comes to is it just me or is it too far away?
Let me fix that, Josh, no worries. Sorry.
All right, hopefully it works better now. Yeah, a major advantage when it comes to fast distribution of vaccines, for example, in the case of a pandemic, is the needle-free administration, as no medical staff is required, and simultaneously the patient compliance is boosted. But more importantly, compared to conventional vaccination, it does not only induce a systemic immunity, but additionally can provide a mucosal immunity by the induction of specific IgA and IgG1 antibodies that can prevent the pathogen entry at the earliest stage of contact and thereby hinder a host infection. But of course, there are also some hurdles that we need to jump. For example, the route of administration itself can be challenging, as the vaccine may have to undergo specific formulation, or it may be cleared right after administration because of short nasal residence time. On top of that, there's the mucosal tolerance. Since our mucosa is constantly exposed to all sorts of antigens all day long, it somehow needs to discriminate between harmful and harmless antigens. And because of that, it can be very challenging to elicit a strong enough immune response via the mucosal route. Therefore, the mucosal vaccination usually requires a very strong adjuvantation. For our strategy to overcome these challenges, we decided to go for a lipopoliplex-mediated dual mRNA delivery, where 1 mRNA encodes for the antigen and the second mRNA for an endogenous adjuvant. You can think of that as a chemokine or a chemokine receptor that is somehow involved in the immune response and recruits more immune cells to the site of vaccination or boosts their migration towards the lymph nodes. I prepare these lipopolyplexes in a 2-step process with a microfluidic system. In the first step, I mix a cationic polymer with a mix of an anionic polymer and our nucleotide to prepare these anionic polyplexes. And in the second step, I take these anionic polyplexes and mix them with an ethanolic lipid stream, and via hydrodynamic focusing, eventually these positive lipopolyplexes are formed. Then, if applicable, I can load the second nucleotide on top of the particle just via pipetting. I use this microfluidic approach as it's really fast, highly reproducible, and has a good upscalability compared to conventional production methods such as the thin film hydration. You may be wondering why I use this second anionic polymer in the core. It's because it allows me a variable loading percentage of my nucleotide. Also, it just reduces the cost in the development of the carrier, and I can exchange the type of nucleotide, for example, from mRNA to DNA, because the particle properties mainly rely on the anionic polymer and not so much on the nucleotide itself. A major advantage of this separate core shell loading is that we get rid of any binding competition between our nucleotides, and we can optimize our shell and our core individually to encapsulate our mRNA. This can be very important if we have systems where the mRNA exhibit very different sizes, because then their binding affinity towards the carrier can be very different. So characterized my particles via DLS. So the polyplexes exhibited a zeta potential of -15 millivolt, and after the coating, this flips to values of around 45 millivolt for the unloaded particles, and it drops again a little bit after the loading to about plus 38 millivolt. I can control the particle size simply by adjusting the mixing speed of the polymers when I do the polyplex formation, and by that I can control the size between about 120 to 220 nanometers. I prove the core shell structure of my particles via cryo-TEM imaging. On the left you can see the polyplexes, and on the right there's the lipopolyplexes. And when we zoom in on some candidates, we can see that the polyplexes are really undefined in their shape, relatively pale, and have no clear structure. But after the coating, we can see this dark lipid shell and clearly the polyplex encapsulated inside. Next, I would like to emphasize why this dual delivery of both mRNAs to the same cell is so important via one carrier, and we cannot simply mix two carriers that each encapsulate one of the nucleotides. So it does not matter for 100% transfection efficiency, then simply all cells will express both mRNAs. However, if the transfection efficiency drops to like 50%, with a separate loading, we would only achieve 25% of the cells that express both mRNAs, while for the dual loaded carrier, it would still be 50%. And this effect becomes more pronounced the lower the transfection efficiency gets. So for example, for 10% transfection efficiency, we would end up with 10 times more double transfected cells compared to the separate loading. And I also proved this hypothesis via transfection of A549 cells. So I used two reporter mRNAs, EGFP and mCherry, loaded one of them in the core and the other one on the surface. Here I did it the other way around. And for control, I mixed single loaded particles together. And I think you can see already some difference if you take a close look at the images. So the cells appear yellowish or orange when they express both mRNAs, since they are red and green, and of course only red or green when they only have one mRNA inside of them. But to make it more visible, I applied some colocalization analysis via software. So what the software does, it looks at the intensity of red and green in each pixel on the picture, and then it plots it in this diagram, and in a perfect system, it would be a narrow straight line, and the steepness of the line would be the ratio of the green to red fluorescence. And I think you can clearly see that for the separate delivery, the distribution is more random and there's no clear correlation visible. However, on the left, we can see this small arm that seems to be separated from the rest of the population, and I think this can be attributed to liposomes that form during the manufacturing of the particles that do not contain any core, therefore only have EGFP loaded on the surface, and that's why they separated from the rest of the population. Recently, I've also tested the effect of the core polymers on our overall transfection efficiency, and for that I mixed 3 different anionic polymers with 6 different cationic polymers and looked at the transfection efficiency in DC 2.4 cells and also the transfection intensity, and by that I've chosen 3 candidates that all have different anionic and cationic polymers for further investigation of the transfection kinetics and also expression of our real vaccine mRNAs and not only reporter genes. And with that, I already come to an end. I'd like to thank you for your attention. I'd like to thank my supervisors, Klaus-Michael Lehr and Brigitta Loretz, as well as the whole DDEL team, our collaborators from Braunschweig, and Dr. Markus Koch from the ENM for taking the cryo-TEM images. Thanks.
Yeah, thank you very much, Alyosha. We have some time for questions. Now it was a very clear presentation.
All right.
But maybe you can comment a bit on the future perspectives, because you still have some time in your PhD ahead of you.
Yeah, a little bit. So as I mentioned, I want to investigate the transfection kinetics. So I want to see if the expression kinetic of the mRNA in the core is different to the one loaded on the surface, because you would think that the one on the surface is easier accessible for the cell to express it faster and better than the one in the core. But that needs to go further or needs some further investigation. And yeah, of course, I also want to investigate our endogenous adjuvant. So right now we have like 2 candidates, and we want to see which one would work better as an adjuvant. But yeah, still some investigations that I need to do.
Excellent. Still no questions? Then join me in thanking Alyosha once more.
Thanks.
All right, our next speaker is Gabriele Bianchi, who is doing his PhD at the Department for Drug Design and Optimization under the supervision of Walid Elgaher. Gabriele.
Morning, everyone. Thank you, Professor Ish, for the kind introduction. Today, I would like to present you my PhD project about the design, synthesis, and biological evaluation of cis-aconitate decarboxylase modulators. Cis-aconitate decarboxylase, ACOD1, or also known as IRG1, is an enzyme deputed to the decarboxylation of one of the intermediates of the TCA cycle, cis-aconic acid, into itaconic acid. It's mainly present in eukaryotes, and in particular in activated immune cells, mitochondria. As said, it catalyzes the decarboxylation of cis-aconitate, which is a tricarboxylic acid compound, into itaconate. The crystal structure was first resolved in 2019 for both the human and the murine variant, and regarding its architecture, it presents as a homodimer in which each monomer contains 2 domains, and in which the active site is located at the verge of the 2 domains. Recent literature highlighted IRG1 as a promising therapeutic target, and its knockout as proving beneficial effects. For example, it can reduce cell infiltration and lung injury in respiratory syncytial virus infected mice, as well as improving the survival in polymicrobial sepsis mice models. Recently, it also proved that the knockout of IRG can suppress multiple tumor growth. And recently, one of the isomers of itaconic acid, which is also the first known inhibitor of ACOD1 cis-aconic acid, proved to reduce ROS and increase survival in human norovirus and Salmonella enterica sequentially infected zebrafish. All of this makes ACOD1 a very interesting target as an anti-infective and immunoprotective therapeutic. So as said, itaconic acid has one isomer, which is itaconic acid, and another isomer, which is misaconic acid. All of those are all endogenous human metabolites, but no interconversion has been proved between the 3 isomers. Because of the Michael receptor moiety, they're all moderate electrophile, with itaconic acid being the strongest of them. For this reason, it's also the strongest KEAP1 NRF2 activator, which is a well-known anti-inflammatory pathway. As said, the itaconic acid is the first natural occurring inhibitor of ACOD1 with an IC50 of 50 micromolar in a cell-free assay, and it acts as a competitive inhibitor via electrostatic interaction within the binding site. In this work, we decided to pursue 3 types of modification: 2-3 modification to probe the space in the binding pocket. Also, we decided to cyclize between position 2 and 3 to remove the Michael-accepting moiety, and we decided to replace the dicarboxy moieties with isosteres. After synthesizing the compounds and sending them to be tested on both the human and the murine variant, we saw clearly that the monosubstitution with bulky and unbulky alkylic group proved not only to retain activity, but also to increase it. Aromatic substitution is also tolerated with regards of the grade of the substitution and the position of the substitution, and also the disubstituted compounds prove to increase activity. For the isosteres series, we saw a retention in activity for the bisamide 16, and so the replacement is feasible with regards of the conformation and the size of the isosteric replacement. The cyclized series proved us to the deleterial effect of a nitrogen in the ring, as well as the relevance of the alpha-beta unsaturation and the aromaticity of the compound. We recently got the IC50 determination in cell-free assay for the best performing compound, and we can clearly see for compound 2, 3, and 4, low micromolar and sub-micromolar IC50. We also got the KI determination, and as you can see, compound 4, 2, 3, and 4 are still our best compounds so far, confirming the IC50s. We also could resolve the first co-crystal structure of one of our compounds into the active site of ACOD1, clearly confirming our compounds as a competitive inhibitor of the enzyme. We then decided to assess the electrophilicity of the newly synthesized compounds. We first performed some quantum mechanical calculation to get this predicted electrophilicity value. We then decided to perform an electrophilicity assay, incubating our compounds with glutathione for 4 hours at room temperature. After that, we performed the AZ-HRMS for the reactions after 4 and 24 hours. And while we could clearly see for citraconic acid the mass of the adduct with glutathione, we couldn't find the mass for our compounds, meaning that they are less electrophilic than citraconic acid, as highlighted also from the predicted value. I would like now to shift gears on another project, investigating itaconate analogs as versatile therapeutics. Recent literature has highlighted the efficacy of 4-octyl-itaconate and dimethyl-itaconate with regards of inflammation and immune response. In particular, 4-octyl-itaconate has proved to activate the previously mentioned NRF2 pathway, inhibiting the release of pro-inflammatory cytokines in lupus erythematosus patients, as well as dampening the lung injury in influenza A virus mice models, and improve the sepsis survival also in mice models. This ability is also shared with dimethyl-itaconate, which also can mitigate the blood-brain barrier disruption in encephalomyelitis-induced mice models. With this in mind, we decided to make some analogs of 4-octyl-itaconate, here 2030, which are the 1-octyl and 4-octyl-itaconate, as well as 31 and 32, which are the octyl amino compounds. And while for 4-octyl-itaconate, we have a very high metabolic instability, as you can clearly see in both mouse and liver microsomes. For our compounds, in particular for compound 31 and 32, and in the half-life, in the metabolic stability. We also recently started to assess the cell viability, the toxicity, and the anti-inflammatory properties of some of the compounds. Here, dimethyl-citraconate and dimethyl-mesaconate. We performed MTT assay on DTHP1 and A549 cell line, and as you can clearly see, for dimethyl-citraconate, we saw a lower toxicity, as well as for dimethyl-mesaconate, no toxicity was displayed even at high concentration. We could also see, interestingly, the reduction of expression of some genes that codify for anti-inflammatory inflammatory cytokines, for example, interferon beta, which is a cytokine that is very much released during viral infections. With this, I would like to sum up my talk. We were able to perform, investigate a novel potent inhibitor of ACOD1, optimize also their physical chemical properties with regards of the electrophilicity. We could confirm the putative binding mode via the competitive mode via crystal structure. And for the second project, we were able to synthesize new itaconate analogs with an improved ADMET tox profile and with an early anti-inflammatory activity. For the next future, we will be exploring new classes of compounds targeting ACOD1 via structure-based virtual screening, and investigate the anti-inflammatory and antiviral activity of the more metabolically stable itaconate analogs. With this, I would like to thank you all for your attention. I would like to thank Dr. Elger and Professor Hirsch for the supervision and accepting me as a PhD. student. I also would like to thank Andreas Cani, our collaborators in Braunschweig and in Hannover at Twincor, and all of you again for your kind attention.
Yeah, thank you very much, Gabriele. We have again some time for questions. Chris.
Oh, yeah, thank you. Really interesting chemistry as well. So can you comment on the reactivity of the compounds with respect to the cis and trans alkenes? And do they form the anhydrides as well?
Yes.
And then the other question was, what kind of doses do you think will be needed for these compounds, and will they be safe?
So you mean for the conformation-wise, or so with regards of the stability of the compounds, they do not form anhydride even with high pH solutions, if this is the question. Regarding the conformation, every conformation has been assessed via NOSI NMR, so I'm sure that every compound has the cis conformation, which is also needed for the interaction with this very electrostatic binding site we have, which is dense of positively charged amino acids. So I hope that answered the question.
Yeah, which is the most reactive, the cis or the trans? I think it's the cis.
Yes, the cis is the most reactive. In fact, they're not reported the values for mesaconic acid, which is the trans form. But in fact, if I remember correctly, they are lower than the one for cetaconic acid. For this reason, also, as said, cetaconic acid, it is in fact the strongest electrophile and the strongest NRF2 activator.
Okay, so Gabriele, it's a very special chemical scaffold and a very small binding pocket. So how optimistic are you about your structure-based virtual screening campaign?
So for the structural, for the virtual screening, we have some idea, for example, an initial refinement within the size, but I also want the compound, we also want the compounds to accommodate and try to find their space in the binding pocket. I think we will not have very much big compounds, as I could have seen clearly from the aromatic substitution series. We cannot grow in some positions, so I don't expect such big compounds, but at the same time, very flexible compounds with maybe two moieties that can bend in a way, so some flexibility involved could then interact with the binding pocket and the 4 very basic amino acids. So yeah, I'm optimistic, very optimistic for that.
Excellent. Yeah, I don't see... Oh, there's one more question, Mario.
Hi, nice compounds, Gabriele. Concerning the first part of your presentation, the ACOD.
Yes.
So you have some compounds that, how I understood that they would bind it, and something happens to the immune response, either gets activated or repressed. Do you have some biological data to see what's happening when your compounds are...
So we are currently planning this. We were discussing this quite recently, so we will perform this kind of assay very, very soon. I mean, this is at the very early stage. At the beginning, we wanted to have some potent inhibitors first, and soon we will perform this assay on zebrafish, for example, but first on full cell, like whole cell assay, especially on the DTHP1 line, which is a differentiated macrophage line, which, as said, is very involved in this immunoresponsive in this immunoresponse. So yes, it's in the plan.
Okay, and you would expect that it would enhance, or?
We would expect that it would buffer the immunoresponse, as the immunoresponse in conditions like sepsis is over, let's say, expressed. Sorry for the repetition, but this is the case. So we wish for a decreased immune response and a decreased release of cytokines that can then drive the inflammation to a next step and increase the inflammatory pattern.
All right, join me in thanking Gabriele once more for his presentation. Our third speaker is Lana Vianney, who is doing her PhD in the group of Julian Hegemann. Lana, the stage is yours.
So, hello everyone. I'm Lana Vianney, and I'm in my first year of PhD for about 6 months now in a microbiota associated natural product Julian Hegemann group. And today I want to present you the main project of my PhD. So a little about this project, which is investigating the antifungal property of the lantipeptide pinancin and its naturally occurring variants. So a little story about this compound first. It's the first and only antifungal lantipeptide that was discovered. And they did like bioactivity test screening, and they see that this compound is active against a wide range of fungi, as you can see here. Yeah, here. It's MIC-HACE, and this compound is produced by Chitinophaga pinensis, and they don't pursue further these studies because they see that this compound also have high cytotoxicity. So the main goal of my PhD is to try to keep the antifungal property and to decrease this cytotoxicity by many means. For this, I need first to produce well the compound and know about the biosynthesis, the gene cluster. So it's class 1 lantipeptide with 2-methylanthonine crosslink with natural product, so with gene-encoded precursor peptide and gene-encoded processing enzyme. The precursor peptide is composed of a leader and a core, and after post-translational modification, you have modified precursor peptide, and the leader can be removed to obtain the RIPS natural product. Here, it's on a paper in 2015. The structure of the pinancin was described, and he have a particularly peer-reviewed moiety. And you have here the gene cluster of the pinancin, which is composed of the precursor and the processing enzyme, and also peptidase, the ABC transporter for the release of the leader peptide, sorry. And you have also the sequence of the core here. To distinguish the class of lantipeptide, you check the processing enzyme. And my pinensis is class 1 lantipeptide, so basically I need to dehydratase enzyme and cyclase enzyme to produce the compound. And a little more about the biosynthesis. The first step is dehydration, so with dehydratase, and it's dependent of glutamyl tRNA, which is a substrate. And it's important to know that when you do heterologous expression, you need to compare the sequence of the glutamyl tRNA of the original producer and your host, expression host bacteria. And because if you don't have the same sequence, there is no recognition, and the dehydration step can be done properly. So after you have some intermediate step, and the last step is cyclization, to have the final compound with lanthonine or methylanthonine. So based on all this information, we did production plasmid based on first duet vector. So in one, we put the precursor and with the cyclase enzyme, and one other plasmid with the dehydratase. And as I said before, we compare the sequence of the glu-tRNA of the original producer, Chitinophaga pinensis, with the sequence of our expression strain, which E. coli BL21, sorry, and is not the same. So we also do a vector to clone the glu-tRNA of the original producer, Chitinophaga pinensis. After doing this vector, we express in E. coli BL21 and isolate the peptide by Ninchek chromatography to obtain a Histec pin-A. And after, in parallel, we did a heterologous expression of the PINT, which is a peptidase containing ABC transporter, to mediate the cleavage of the leader and obtain the lantipeptide. And after HPLC purification, we have the final compound. Not totally the final compound, because you have some 4 amino acids here. And here, it's just to show you that when we don't put the vector with the glutear RNA of the original producer, you don't have a complete dehydration. And after the treatment with the Pinchy, we obtain the final compound, but with 4 acid amine in addition. So what was done is to generate variant to allow the complete core release with commercially available protease. And after we check if it works, so it works well. We obtain the compound with the pyruvyl moiety. And the collaborator, Machtadler Veracolici, showed that not only the mature pinencyne is active against a model of fungi, but also the intermediate compound. So you see here that there is no complete pyruvyl moiety, and you have an activity. So maybe not as well as this one, but we have an activity. In progress now, I try to optimize the pinencyne production to have good yield to do my next step, which is in mode of action studies and structural activity relationship studies by mutation and something like that. And another part very important of my project is the genome mining. Why? Because genome mining might reveal variant with reduced cytotoxicity. And we did the study of genome mining, and it revealed 3 major subtypes and 3 additional singletons. So the 3 major subtypes and the singleton. And I want to attract your attention in the clade tree. Why? Because Elizabeth Kingia are an emerging class of antibiotic-resistant hospital pathogen, and it could be a very an important side part of my project, because we can wondering the role of the pinencyne on the pathogenicity of Elizabeth Kingia. So we try also to produce the clade 2 and clade 3 in E. coli, and it's worked well. And from April, I have with me Amélie Rowe, that come from Polytechnique X in Paris, to work with me on the the pinencyne-like biosynthetic gene clusters, so the variants. And we select some various DSMZ strains. And we did agar overlay assay against the fungi model. And what is interesting to show you here is that after 6-day culture in the same condition, we have some variants that are active, show a little activity, but not the pinencyne that is the original producer. So it means that the variant produced in more quantity or are more active. And after 10-day culture, we have a little activity for the pinencyne and also activity for some of the variants that we don't have also activity before, like for Chitinophaga japonensis here. Now in progress, we need to purify the pinencyne variant and also optimize the production, and next do the activity and cytotoxic test and do a lot of comparison with the pinencyne compound. And I wanted to say thank you to my group, particularly Julian Egeman, my lab mate, the AMEC group that hosted us in their lab and to helping me a lot with the experiment, and Anna Hirsch to be my scientific co-advisor. Thank you.
Yeah, thank you very much, Lana, for a very clear presentation. I see one question from Lone.
So thank you very much. That's a super cool study.
Thank you.
So this is a very technical question. So in your last assay where you've selected different species organisms that you by bioinformatics know likely produce your rib, do you actually know that the compound was produced under the culture conditions? So the lack of inhibition, is that lack of activity or is it lack of production?
We search actually in the mass spectrometry data that if the compounds are produced or not. We find some, but not all. And we find also that on the variant we have also other ribs. And so we check, and we don't know if it's really the variant of the pinentin or not that are active. So it's in progress research.
Mario, do you have a question?
Yeah. It's now on. I just wanted to ask about your last essay where there are the inhibition zones on the agar. The culture, it was for 10 days. I want to ask how is this done and why is the effect seen after 10 days?
We culture in a big flask 500 ml of media. It's homologously produced. It's not heterologous production. I just buy the strain by the GSMZ, and we cultivate in various days to see if there is a difference or not. And after we just extract, we use XAD resin. And after we do methanol extraction, and after we have extract, and it was just a crude extract that we test first. And
the inhibition you saw after 10 days, right?
For some compound, it's just after 10 days. Okay. So maybe it's more active at 10 days of cultivation, or more produced.
Okay, thanks.
Yeah, maybe some other PhD students or postdocs would like to ask questions, especially in this session. We have some time. There's one more speaker to go, but I have one question. You mentioned that you will now do structure-activity relationships. How will you generate the derivatives, all by heterologous expression and variations?
We want to make some variants. Some mutation, because with the RIPS, what is good is that you just can replace the core part, like by mutation, and you have a different compound. So let's see, we just want to start with some mutation and see the activity of the different mutants and the structure related.
Excellent. The best of luck with that.
Thank you.
So, many thanks, Lana. All right, our final speaker, and I hope I get the name more or less right, is Amay Ajay Kumar Agrawal. He's doing his PhD in the group of Olga Kalinina.
So, good afternoon, everyone. First of all, I would like to thank the organizers for giving me this opportunity. Today in this talk, I will dig a bit into the biosynthetic potential of highly diversified phylum, that is Myxococota. First, I will start with the basic introduction about natural products. Natural products, as the name suggests, are basically small molecules produced by plants, microbes, fungi, and other microorganisms found in nature. Unlike primary metabolites, they are not directly involved in growth and development, but they help the organism to survive, to adapt to the surrounding environment. And one of the most famous examples of natural product is the penicillin antibiotic, which is derived from the fungus Penicillium rubens. And apart from antibiotic activities, these natural products also exhibit diverse biological activities, including anti-cancer, antiviral, anti-parasitic, etc. In this talk, we will talk about the Myxococota phylum, which is a rich source of natural products, and the natural products coming from this phylum has high structural and functional diversity. Over the last few decades, many different studies have used metabolomics and other laboratory procedures to unearth the myxobacterial diversity to some extent, but a large fraction of it still remains hidden. Thus, in our study, we introduced 129 new genomes belonging to the Myxococcota phylum. And these 129 strains, they were isolated from different type of samples like soil, sediments, and decaying plant materials. And all these, some of these samples, they were collected as a part of citizen science project called Microbialics. And most of our strains, they exhibit exciting morphologies, as we can see in this image here, captured by our colleague Ronald. Next, after isolating these strains, we sequence them, assemble them, and then use them for further downstream analysis. After assembling the strains, our first task was to identify the taxonomy of our strains. And for this, we used current state-of-the-art approaches like 16S or the UNI values. But what we found here that most of our strains, they were not well resolved at family and the genus level. So to overcome this, we saw that some of the previous studies have used POCP, that is percentage of conserved proteins, and shown that it effectively works for other taxonomy levels. Thus, our goal for this part of the study was to find the POCP cutoffs for family and genus level, and then use these cutoffs to perform the taxonomic refinement in Myxococcota phylum. So to achieve this, first we downloaded the type strains belonging to the Myxococcota phylum, and then build this species level pairwise POCP matrix, as we can see here. Next, to go from this species to family level POCP matrix, let's assume that the species S1 and S2, it belongs to the family F1. And to calculate the value of this cell, we took the average of all the cells involving S1 and S2 except the diagonal. And in the similar way, we calculated the value for other families as well. And we also arrive, using the similar procedure, we also arrive at the genus level POCP matrix from the species level POCP matrix as well. Next, this is the heat map showing the family level POCP matrix based on the type strains belonging to the Myxococcota phylum. Now, in this heat map, the values along this diagonal, it represents the POCP value between the same families, while all these others, while all these other cells, it represents the POCP values between different families. And what we found here was that for all the cells along the diagonal, we had the POCP value greater than 50%, while for all the other cells except this one, we had POCP value less than 50%. So based on this, we defined the POCP threshold to be 50% for the family level. And for this cell, which has POCP greater than 50%, we also suggested to merge the two families belonging to this cell. Similarly, we defined the POCP threshold 76% for the genus level as well. And next, we used both these thresholds and applied it to our in-house Myxococcota strains. And this is the final family and genus level POCP matrix of our 129 in-house strains. Now, the total number of families and genera in the current Myxo taxonomy is 10 and 31 respectively. And based on our defined POCP thresholds, we introduced 90 new families and 63 new genera involving some reclassification suggestions to the current Myxo taxonomy. Thus, after applying our thresholds, the number of families and genera in the new Myxo taxonomy will be 28 and 94 respectively, which is quite exciting. And finally, this is the phylogenetic tree built on the basis of 129 in-house strains plus the type strains. And the blue leaves in this tree, it represents the strain coming from the new families that we have introduced in our study, while these green leaves, it represents the rest of the in-house strains. After looking at the taxonomy, next we wanted to explore the biosynthetic potential of Myxococcota phylum. And for this, first we identified the BGCs in our in-house strains using AntiSmash. And we also downloaded the public BGCs from AntiSmash DB belonging to Actinomycetota as well as Myxococcota phylum, and the public BGCs from MyBig. And this is the bar plot showing the total number of BGCs across all the different data sources. Now, in this bar plot, we can see that public Actinos has the highest number of BGCs, while it also has the highest number of strains, which is represented in this bracket, while the number of BGCs in our in-house Myxo strains is around 3,700 from 129 strains. And this is the stacked bar plot showing the distribution of these BGCs into important BGC categories. And here, if we compare our in-house Mixo with the public Mixo and public Actino, we can see that it shows quite a different distribution, which means that our in-house Mixo might contain a lot of novel secondary metabolites. And to further verify that, we clustered all these BGCs using BigSlice to identify the gene cluster families. This is the table showing the number of GCFs across all the different data sources. And next, we did the Venn diagram analysis to see how many GCFs are shared across different data sources. And in this Venn diagram, we can see that there are very few GCFs which are shared between in-house Mixo and MyBig, while there are higher number of GCFs which are shared between in-house and public Mixo, which is kind of expected. And when we look at this in-house Mixo Kokota GCFs, we can see that there are larger number of GCFs which is not clustered with any public data. And similar results were obtained when we compared our in-house Mixo and public Mixo with public Actino as well. So overall, 83% of our in-house Mixo Kokota GCFs are novel and not clustered with any public data. And one more important result that we found here was that although the total number of our in-house Mixo strains are quite less as compared to the total Actino strains, but still the total number of our in-house Mixo Kokota GCFs is roughly 1/10 of the total Actino GCFs. Thus, our in-house Mixo Kokota comprises a huge biosynthetic potential which can be explored further. Next, we wanted to see that which families in our in-house Mixo strains contributes the most towards the GCF diversity. And for this, we plotted this bar plot showing the average number of BGCs and GCFs across all the different families. And in this bar plot, if we focus on this new family N, we can see that although it has a comparable genome size as compared to other families, but it has the highest average number of BGCs and GCFs as compared to other families. So this seems to be a very interesting family. And in fact, in the latest publication by Professor Roll's group, they have named this new family N as Pendulus Poraceae, where they showed that the strains coming from this family, they exhibit distinct sporulation behavior, which was previously unseen in any myxobacteria. And also the natural products coming from this family shows unprecedented chemistry and enzymology. Further, we also created a database called ABC Mixo to explore the BGCs and its associated functionalities. So this is the screenshot of the database showing the BGCs and its associated data, while this is how the GCF visualization looks like in the database. Next, to summarize this presentation, we introduced 129 new strains to the Myxococota phylum. Next, we overhauled the current taxonomy of Myxococota phylum using POCP. And in this process, we introduced 19 new families and 63 new genera to the current Mixo taxonomy. Next, by clustering our in-house BGCs into GCFs, we increased the current Myxococota GCF atlas by 4-folds, wherein we found that 83% of our in-house Mixo GCFs are novel and not clustered with any public data. And all these novel GCFs, it represents new potential scaffolds for drug development in future. Finally, I would like to thank everyone who is involved in this project. And specifically, I would like to thank Professor Kalinina and Professor Mueller for giving me the opportunity to work on this project and supervising the whole project. And next, I would like to thank everyone for your time and attention.
Yeah, thank you very much. Another very clear presentation and the fourth talk that's on time. So congratulations to the 4 speakers already in advance. And we have some questions. Lone.
Super cool talk. Thanks a lot. So you've discovered lots of potential novel chemistry, and that's obviously what we're looking for, a lot of us. So I have really 2 questions for that. One is related to how are you going to prioritize? So if you've quadrupled the unknown chemistry, I realize that you're referring to a publication where you're looking for novel. So that was the first question, how are you going to prioritize it? The second question is really, and this is my ignorance on Myxococcus, is do you have expressions hosts? Do you rely on production in the native organism? How are you going to dig into the chemistry for these unknown BGCs?
So regarding the first part, for prioritization, we saw that in our bar plot, we saw that there were some families which are quite interesting. So first we will look at the BGCs and the GCFs for these specific families. And we will use kind of some certain metabolomic approaches to kind of see which one seems to be interesting one. And as we saw that the Pendulous Sporaceae paper, which was already out by them. So similarly, we can either look at other GCFs from these families, or we can also look at other families which are also quite interesting in this case. And regarding the second part, actually I am a trained bioinformatician, so I'm not sure much about the biology part, but we have our colleague Ronald here who can answer this question. So maybe we can do it now, or we can, yeah.
He's right at the back.
I think for the second question regarding the host, we're actually developing several mixed bacteria as a host, so it's still ongoing. Thank you.
All right, that's a good example of teamwork. Do we have more questions? Yes, please.
Thanks for the nice talk. Do you know this public data set you presented? How many of these strains can actually be cultured in the lab? Can they all be cultured, or is it just?
No, it's all the culture data that the public data sets you are.
Public data, yeah.
Yeah. So the anti-SMASH DB, it's so the database that we are using. It apparently takes all the genomes from NCBI and identifies the BGCs. So.
But can all the strains be cultured, or is it just basically sometimes you can get the DNA, but you are not able to culture the strain in the lab?
I'm not sure about that one.
Okay.
But yeah.
Okay, Rolf will help out.
I'm getting nervous here. So the whole point of this is they are all cultured.
Yeah.
We can grow these strains. This is not some metagenomics blah blah. This is real microbiology.
Okay, it seems that the most urgent questions have been answered. There will be plenty of time in the lunch break and the coffee break and the barbecue to ask even more questions. Do join me in thanking all of our speakers of this session for a great job. And because they were all on time, we actually have an even longer lunch break, so plenty of time to visit the posters. Please don't forget to vote online using the star system, 3 stars for the best posters. And then we will reconvene at 1:30 for the next session. Thank you very much.
Hello. Good afternoon, everyone. Welcome to presentation session 2. My name is Olga Kalinina. I am a group leader for drug bioinformatics in HIPS, and I will be chairing this session. Our first speaker is Emma Szymanski. Emma is a chemist known for her work identifying unknown organic compounds, in particular pollutants, and an advocate for open science. Emma graduated as a chemist and environmental engineer from the University of Western Australia and spent a few years working as an environmental engineer, after which she moved to Germany, to Leipzig, to the Helmholtz Center for Environmental Research, where she did her PhD, after which she moved to Switzerland with a Marie Curie intra-European postdoctoral fellowship. Currently, Emma is an associate professor at the University of Luxembourg, where she is the head of the environmental chemoinformatics group. Of particular achievement, I would like to mention that Emma started a critical assessment of small molecules identification contest that provided researchers with information about unknown organic compounds and challenged them to use automated computational tools identifying unknown compounds, which is in particular dear to my computational heart. So we would like to welcome Emma here on stage. Thank you very much.
Thank you very much for the introduction, and thanks to all of you for coming here. I hope you had a good lunch. I actually have very good material to close with a joke just before a break, but this time I'm right afterwards, so I haven't included that joke today. But I'll show the pictures anyway. I feel like I'm a bit of an outsider coming to this conference, so I'm very honored to be here. I hope I can also give you an entertaining talk. In a way, I could give the punchline, I do drug discovery, but in water, and not the way that many of you do. So the talk today is going to be environmental cheminformatics. How can we find meaningful hits amongst millions of chemicals? Because this is kind of our challenge from the environmental perspective. So either the bio I gave was completely out of date, or I don't know how that happened, but so I've been a full professor now since the end of September, which was also a milestone for all of those of you who are aiming for something like this. It's good that we finally reached this stage as a group. I do have a lot of pictures today, so, oops. That was funny. The slides for my talk are actually available on Zenodo to download. I go through a lot of pictures, have a lot of hyperlinks in there. If you want to download them and go through in a bit more time, then please do this afterwards. All right, so environmental cheminformatics, what is it that I do and my group does? Basically, we look at the environment. We take samples from the environment, or now also from humans, biosamples. We then process these with high-resolution mass spectrometry, generally with an Orbitrap. This is where then the cheminformatics comes in. How can we make sense of the data coming off the instrument? With the eventual aim quite similar to yours, but in a different context, how do we identify chemicals and their toxic effects? Because we're trying to find the chemicals that are of concern for the environment, or now also in the context of disease. So the focus of my group is really on the middle part, the high-resolution mass spectrometry and the cheminformatics. We develop a lot of different software, and yes, as I was introduced, also a lot of open software. I haven't put a strong open focus on it, but actually everything we develop as far as it can is open and available to use. Same with my slides. I'm going to basically walk you through all of these developments in my talk today. And for those of you who know the software R, then you'll know where the hex stickers are coming from, because this is basically what we do in R with all of our packages. So in the environmental context, what do we do with non-target high-resolution mass spectrometry? Firstly, we start off with the experimental design. So this will either be lab experiments, we can do treatment plants, we can do just routine monitoring, we can do now biomedical studies as well. You can do the sampling, and then you've got a sample archive. We can then do the analysis like we do on the non-target machines, and then go what we call the digital archive. So you've basically got your snapshot and building up a picture over time. Then comes the fun part, the informatics side. You've got the pre-processing to get an overview of what's in the samples, prioritization according to different patterns or different questions, and then finally the identification at the end. Just to give you some examples of where we apply these methods in the more environmental context, this is what I did in my time in Switzerland. We've got, for instance, the real-time monitoring of the Rhine River. And if you look at the bottom here, we've actually had daily samples. This is between 2014 and 2016, but it's still going on. And what you can do with the pattern analysis is we can, like on the top you see here, you can start to see chemicals that are only appearing every so often in the sample. So this was appearing once a year. Those peaks, this is associated with 0.6 tons of that chemical. So this is a huge amount of chemical that's all of a sudden coming into the Rhine River. So you can see the first one, 0.6 tons, the next one, 0.1 ton, 0.4, 0.2, so cumulatively this is well over a ton of material. And in actually identifying this and finding it, you can then go back to the company and say, hey guys, we've found this, can you please stop putting this into the river so that, you know, the people in Stuttgart are not drinking this compound because they're taking their drinking water from the Rhine. The interesting reverse of that is this bottom case, that you actually have these signals that are occurring all the time, and then all of a sudden they'll drop out for 6 weeks. And so why does this signal suddenly disappeared? And what was happening there is we actually had a production break in the company. So again, this pattern stood out, and you can see in the middle of 2015 they actually put in a mitigation measure, and the chemical's still coming, but it's much lower. So this is again a much easier problem for us to treat now that the company knows how they can avoid putting this into the Rhine. We've also got this is hopefully an example that will speak to a lot of you. We can also just get an overview of the surface waters in Luxembourg, what's happening with the pharmaceuticals in the surface waters. And this was a really interesting study, my postdoc Randolph and my PhD student Adeline. In the purple you see 2019, and in the green you've got 2020. Luxembourg is very special. We have a lot of cross-border workers, so when everyone closed down their borders, our population during the day halved, because our population basically doubles during the day and halves again at night. Everyone was at home, and you can see the pharmaceutical concentration. The green is consistently lower for almost all of the pharmaceuticals. So for instance, nicotine, it's less than half in 2020 versus 2019. And then we've got other ones like iohexol. All of the hospitals stopped any of their imaging and diagnosis while they were treating the COVID patients. So this signal actually dropped out completely from one of our most intense signals the year before. We also look at chemicals like pesticides. So this was Jessie, one of my first master's students. And the challenge we obviously face in the environment, like you do in the human body, all of these chemicals can metabolize and transform in the environment as well. So we're not just looking for the parent, we're also looking for their different potential transformation products and trying to reconcile them with our data as well. You can also start to get an overview of where these chemicals are occurring throughout Europe. So this is an example from the Black Sea. You can see we've got a drop-down menu here where you can pick a chemical, and then you can basically build the map and see where these concentrations are happening, and you can see this big red dot here. This is a city that doesn't have any wastewater treatment, so this is going untreated into the Black Sea, and then you can identify the hotspots where we have to implement some actions. And this is actually, it's a platform where not only do we look for the mass, but where we've got the fragmentation information, you try and look for the fragments as well, so get the identification as good as we can. You can also, over time, start to get a distribution of the chemicals. So once we know there are chemicals of concern, are they actually going to be in the water? Like you see in the middle here, you can see chemicals like atrazine, terbutarazine, they're all coming in the water, hardly at all in the fish or in the sediments. But then there are some other chemicals like this chlorhexidine, it's very intense in the sediments, but we don't see it anywhere else. So of course, you wouldn't bother looking for that one in the water. And then you can also see which ones are actually accumulating in the fish and then becoming also a problem for consumption. So how do we make all of this happen? A lot of this is through collaborative developments, and I just wanted to walk you through the main ones that you see on the left here, bit by bit. So obviously, a lot of you know mass spectra, they're the fingerprint of our chemicals, so we also work on mass bank. With Anjana, who was a master's student, she's now a scientist in my group. She was responsible for the first Luxembourg records in mass bank, but also we got the first organometallic records in mass bank as well, because this from a cheminformatics perspective was not particularly non-trivial. So we're doing a lot of work. This is both open source software and the open data. The entire data in mass bank is available for use. We also gather a lot of expert knowledge from the environmental experts in the community, because everyone's working on different chemicals, so they all know the most about their chemicals. And we maintain the suspect list exchange to basically capture this knowledge and exchange it with other databases. The website itself looks very simple. We published this a couple of years ago, 97 authors on there. I did actually ask over 100, but a couple of them just wanted to be acknowledged, so we almost got our 100 author paper. By now, we've got more than 100 contributors to the lists as well. How does this work? So we get suspect lists in a variety of formats and colors and every single possible form you could almost imagine. What do we do in the suspect list exchange? We kind of standardize this into a format that most people can read. We then deposit these on Zenodo, get an individual DOI for these sets. This also gives us the statistics, so we can tell what are our most popular lists. Believe it or not, cosmetics has been a total hit. We had no idea. And then once they're on Zenodo, we actually integrate this within the Norman database system. So that feeds SUSStat. This is actually the database that is feeding this digital sample freezing platform. So this drop-down menu, all the standards in here, are actually coming from our lists into the main database and then into the screening. So this is the way we're transferring information from one resource to the other. And then what we also do is we make sure it's in some of the other major open databases that a lot of researchers use. For the environmental community, this is CompTox in America, the US EPA, and then also PubChem, which is not just environmental audience, but obviously a very big biomedical audience as well. And you can see with PubChem, we don't just give them the chemicals, but we also give a lot of this information content as well. For us, the software METFRAG is the way we connect a lot of this information together. So METFRAG is not a single software, the in silico fragmentation, which is what it was originally designed for. But we also now use this to connect in MassBank, in the suspect list exchange, CompTox, PubChem, all of this extra data. All of this we consider then in our candidate ranking. And so, for instance, if we've got the chemical nicotine, obviously there's a lot of candidates with that molecular formula or the mass. But with all of this extra knowledge, you can actually prioritize it, and it's pretty clear if we've got a human sample, and especially if they're a smoker, we're going to find nicotine in there and not most of these other candidates. So the grand challenge that we're dealing with in our research is actually that our chemical space is too big. So CAS, which is one of the largest databases out there, is currently 219 million chemicals. PubChem is 118 million. When we first published METFRAG in 2010, PubChem was 25 million. So that's several factors higher now. ChemSpider is already at 129 million, and even CompTox, which started as a very small database, has already hit 1.2 million chemicals as well. What does this mean? If we would screen a mass or a molecular formula in PubChem, for example of nicotine, we end up with close to 3,000 candidates. What does this mean? We have very few candidates with high information content that we know that we're likely to find, and we've got this very, very long tail of candidates with low information content, and we're getting prohibitive runtimes. So we were looking at, with our software processing, measuring a sample in 20 minutes. It's taking you 2 days to do the data analysis. So we definitely wanted to do something about that. And what was the key for us is not using the entirety of PubChem. A lot of this is coming from patents. They're not actually produced in an amount that we'll ever see in the environment. So how can we actually look at PubChem and get the informative subsets out of it that we're interested in? So you can see here in yellow, I've highlighted the ones that are interesting to us as environmental researchers. We've got the agrochemicals, the pesticides, drugs and medication information, the pharmaceuticals, food additives and ingredients. We've got pharmacology, biochemistry information, safety, toxicity, use and manufacturing information. So if they've got use and manufacturing, this is also produced in a significant amount. What it also means is if they've actually got any information in this category, it means we actually can go into PubChem and look up exactly why that chemical has been mentioned in that category, so you can also begin to validate these hits. You know, is this actually meaningful in my context? Because it could be that we have use in manufacturing, but if they say it's banned in Europe, maybe we shouldn't be finding it in our samples, or if we do find it in our samples, why is it there? Where is it coming from? And it does actually come, we do actually get cases like that. From the exposomics point of view, we've got the associated disorders and diseases. Obviously, if we're looking at diseases, we want to know any chemicals that have been associated with this disease and are there any studies on it, and you'll actually get to the table that points you straight away to studies that have documented these chemicals. And for the metabolomics side, we also put in the metabolomics pathways so that we get the natural transformations that are happening there. Sorry. So all up, PubChem Lite, instead of being the 118 million that PubChem is, when we squish all of this together, it's actually a database that's about 375,000. So in terms of efficient workflows, this is a much easier thing for us to work with, and we achieved our aim straight away. Our 20-minute sample acquisition, we can now process in well under 20 minutes of data. So that's what the difference makes with that optimization. We're actually updating PubChem monthly, so PubChem itself is changing daily, the annotation content weekly. We actually build PubChem Lite every week. We have a continual monitoring, but it doesn't change that much, so we only give monthly updates. These are all on Zenodo. And what that means is whenever we have an update, you can see we're already at version 33, we can also directly integrate this into our other software pipelines. So what does PubChem Lite mean? It means we've got fewer, much more relevant candidates, but not only do we have fewer candidates, we also have the context. So I'm going to, at the end of the talk a little bit more about literature and patent values and how these help us. But we also have an annotation count score, so we can sort them according to the subsets as well and even do some more filtering based on this. So I showed the workflow of the high-resolution mass spectrometry and how we go all the way from sampling to the identification. I'm not going to go into great detail today, but we do have workflows. This is Rick Helmus, who's a PhD student in Amsterdam. I'm fortunate enough to be his external supervisor. This was actually a completely self-motivated project, but he has got this pipeline that's basically going from the beginning, data acquisition and pre-treatment, all the way through to this annotation. So we build little bits and pieces of the workflow. The great thing working with Rick is as soon as there's a new thing involved, he will integrate it straight away into the pipeline that we can then use also in the pipelines that we're using in our lab. So what about transformation products? I already mentioned them a little bit, that we have a lot of these in the environment. So I bring up again the example from Jessie, because she actually motivated the work that Emma has been doing that I'm going to show. Because we have a lot of, sometimes, I mean, in this case, we were lucky, we see the parent and we also see the transformation products. And if you can see with the colors, you'll see that actually the ones in the middle, they're colored blue and not green. So these are actually happening at a different month, so they must be coming from a different chemical. So, but we have the cases where we only see the transformation product and not necessarily the parent. So how do we actually get this information and interlink it as well? Another thing we've been doing is, we put a lot of effort into breaking PubChem down to a very small subset, PubChem Lite. The problem is when we do a transformation product prediction, we expand it straight back out again into the millions. So if we do one set of either human or environmental predictions, we're already at 1 or 2 million. If you do a combination of human and environment, which is kind of what we would see in the wastewater treatment plant, we're already at 11 million possibilities. Do a few generation of environmental transformations, you're already back at 82 million. This is almost the size of what PubChem was. 2 to 3 mixed generations, and you're also back to the size of the original PubChem. So our problem here is all of these predictions, combinatorial explosion, we don't have enough data to limit the predictions to what we really see in the environment. So this is where the work of Emma Palm in my lab is coming in. We've been working a lot with saving data in a fair way to get into PubChem, but we're also trying to look at what's the text records that are in PubChem and other resources that we can harness and curate very easily to expand this knowledge. And there's a huge amount of knowledge in PubChem coming from the HSDB, so a database that was maintained by the NIH, hand-curated by 40 scientists over many years. And this is all text-based. But they're also running text mining algorithms over this, and you can actually do a very simple screening and a quick validation. All those ones in green are actually valid transformations. The one in orange has been incorrectly recognized. It's only a partial recognition. And the thymidine is in a completely different context. This has no actual relation to the acyclovir. So what Emma did is she's actually designed a little Shiny app that automates this process, retrieves the data, visualizes the reaction, so you can very quickly go, yep, this one's real. No, sorry, this one doesn't make any sense. Already exports it in a way that we can update it on Zenodo and automatically import this into our workflows. So what we've been able to do with this is, so the biotransformer, the predictive system, had about 3,000 reactions. We've been able to more than double this with all of the data set that we've gathered. And again, it's public in PubChem, but we also archive it on Zenodo. This means that Rick can pull in this data into Patron, and this means that we can basically live always look at the latest reaction set in our software as well. And the way that this works is we basically start with Patron. If you would go through your sample, we can have a look at the different hits and say, okay, this one is looking really interesting, and it's a pretty high concentration. Let's see if we can find any transformation products related to it. So you can feed in basically all your tentative identifications, predict the transformation products. And then once you've got these transformation products, do a second layer of screening, and you say, okay, it's not just this one hit here, but we've actually got 3 related chemicals that are associated with it as well. So already you can use the first preliminary hit to cross-annotate another 4 or 5 peaks, or sometimes more, sometimes less. And then you also get the building of the tree, and then you can see how this metabolism process is actually happening. So I hope I still have a little bit of time. because this is the work that we're currently doing. I had a meeting actually on the patents just before coming here. This has ended up being a side project that was, it was a very interesting package. It has now become one of our most over-designed R packages we've ever produced, because we got so much interest in it. PubChem were then incentivized to actually provide it in a much more efficient format that meant we didn't have massive downloads, but super trivial downloads. So, one thing, I mean, I had in the title, how do we find the most relevant hits? And one thing we're also doing as a group is challenging what's happening in environmental regulation. So, any of you who have heard of the perfluorinated substances, I mean, or seen the film Dark Waters, you'll know there's a lot of attention coming to this PFAS. And there's been a push for a new regulation that has redefined the definition of a PFAS as anything containing a CF2. And hopefully for you as an audience, you know the relevance of a CF2 or the CF3 as a binding group. This means it's actually impacting a lot of drug discovery experiments. When we upscaled this new rule to PubChem, it's actually hitting 6 million entries in PubChem. So then we're trying to challenge, like, okay, do you really want to regulate 6 million compounds, and how are we going to deal with this? So we created quite a stir when we presented this. How did we do this? We actually automated it into the PubChem PFAS tree. This one's actually updating weekly on PubChem. And if you actually go into PubChem, we've actually got 22 million compounds in PubChem that are containing a fluorine atom. And we've gone to the OECD definition and we've broken this down. So organofluorines, you'll see that's 21 million. And then of the subset containing this OECD PFAS definition with the CF2, we've got 6.8 million. And then we've actually broken it down with the salts and mixtures as well. If we take a closer look at this, you'll see 700,000 of them just contain a CF2 group, and a whopping 5.9 million, almost 6 million themselves. These are containing the CF3 group, and this is the one that's really hitting drug discovery in the pharmaceuticals, because that's an awful lot of chemicals that are also being potentially marketed. And then if we actually look at what's the classic PFAS definition of a chain longer than a CF2, CF3, we've only got 200,000, which is still an awful lot to deal with, but it's also not 6 million big. So one of the things we've really struggled with over time is to kind of describe the complexity of the data, or just the immense amount of data we're dealing with. So what we actually did, so Dagny worked on visualization. When I was trying to actually articulate what we're doing, I met Jamie, who's a musician, and he was like, okay, I really want to put this to music, because he could see I can make this story work for you. So bear with me, this is an animation. Hopefully the sound is going to work as well. This is just going to trace you through our chemical past, present, and future. So now you can see there's a drum beat in the back. This drum beat is every year. What you can see here coming up, this is the patent count for these chemicals over the years. So you can see PFOA we don't even have registered as patents yet. So in 1962, Rachel Carson documented the problems with DDT in a book, Silent Spring, basically saying the river is silent, the spring is silent because the pesticides are killing all of the insects. A few years later, 1965, this is when CAS first started releasing the CAS database. So you can see here on the black line that's just coming up, this is actually how CAS is expanding over time. If you have a look at this axis over there, it's scaled to 20 million. This is the number of chemicals being added per year to CAS. So you can see already we're in the 80s, not terribly many patents. CAS is still very small. We're already getting the first big chemical disasters. This was one in Italy. And in 1980, there were a couple of disasters that really kicked it off. Here was one in India. And then many of you will probably know the one that affected the Rhine, this Sandoz chemical spill. This is actually what stimulated the daily monitoring of the Rhine that I showed you before. The Dark Water scandal, this actually started happening in the 1990s. So that's the film Dark Water. It took them 10 years before they even started legal action. Have a look at the size of CAS. It's still extremely small. So even though they had the documentation of the PFAS, it took them 10 years before the legal action started. And now the chemical numbers are only just starting to expand, right? So we know PFAS is a problem. We know DDT is a problem. The first restriction of DDT came in 2000, 40 years after it was documented. And now you can start to see what's happening with the patents, what's happening with the chemicals. The number of chemicals being added to the databases is going through the roof. We've only got the second chemical restricted. PFOS was only restricted just before 2010. And already we know there are a lot of chemicals that are still problematic. Have a look at the patent numbers. We're going into the red zone. The numbers of patents are going through the roof as well. Dark Waters was only just released in the late 2010s. River Order disaster happened only last year, and then the PFHXS and some of the other PFAS were restricted only last year as well. So it's like we've managed, just out of these chemicals, we've managed to restrict 4 of them, yet we're getting 20 million added per year. Rachel Carson, when she wrote her book, she was concerned that 200 chemicals were being added a year. So I won't take too much longer. So what we started to do is, you see the systematic stripe pattern. You always see this green and it's going to the red. We can do this for the patents. We can do this for the literature as well. We can also do this for groups of chemicals. So you can see here, this is a regulated class of PFAS. Is this as a class also increasing in the patents? And the really interesting thing about this, and when you're doing informatics development, it's always cool to have some test cases, because we have the really well-known things like caffeine and nicotine. That's where I can build the good joke if you're just before the coffee break. But then we also have our test cases. So we have the test case CID1. It happens to be a natural compound, and here you can see for the natural compounds, we see quite different patterns to the well-known chemicals. And then here, my favorite test case, I just put in 1, 2, 3, 4, and we went, hang on a minute, that's a totally different pattern to what we've seen before. Because you can see it goes green at the end, and it peaks much earlier. And then when we did the same for the literature, it peaked even earlier. So we can see the same trends are happening. So it turns out that galopamil was documented. It was a really cool drug. It was going to be, I can't remember what it was going to be used in treatment. They put it into clinical trials. It had very severe side effects, and they took it off again. So that explains why it all of a sudden had a really good peak and then completely disappeared out of the literature, because they switched to other drugs. So the lesson from this, and this is very close to the end, what we want to do with the patents is start to see, can we see those chemicals that are up and coming and catch them before they become a problem? So this is Daniel Zahn, he's at UFZ, and he gave a talk at CTAC last year, and he was already flagging the ions that are really concerning for him that he's seeing in the wastewater that we can't treat. And when we looked at a couple of the ones I've got in the red box there, we did this live during the presentation because she had the package already. We did tetrafluoroboric acid, which is the one he's got there, and pattern was kind of normal. But then when we went to the lithium salt, this was the first one we've seen only go orange towards the end and not go back to green. So these patents are going through the roof, primarily from China, and these are associated with electric vehicles. So we've got our next PFAS disaster coming right there, because these chemicals are going into the wastewater, and we're already starting to see them appear. So with that, a little bit of detective work, hopefully a little bit of interesting anecdotes about our research. I hope I've been able to show some of our developments that we do in my group. If you're interested in the video or the soundtrack, it's on Vimeo and also on SoundCloud. We've got the QR code there. You can also download today's slides to get some of these hyperlinks on Zenodo, so the QR code is there. Thank you to my group, all of our collaborators, the community that helps, and thank you for listening. Happy to take questions.
Thank you very much for an exciting and very artistic talk. Do we have questions? Oh, right.
Yes, first of all, thanks for your interesting talk, Emma. Always happy to see work done in the environment. Actually, I have a question for one of your first slides. It was slide number 4. Maybe you can answer like this. You had the 2 graphs, and if I understood correctly, both of them refer to the same substance, the PPA, right?
I'm just getting to slide 4 on my phone so I can see it at least. They were 2 different compounds, yes.
Okay, 2 different ones. Okay.
So they were, because they're almost the reverse. So they both came from industry, but yes, one is PPA and the other one is TCP. So very different structures. Yeah.
Perfect. Thank you.
Yeah. You know, the cool thing about the mass spec is you can trace back with the mass and actually find where they're coming from. So, sorry, I saw another hand come. Yeah.
Thank you very much. I was just wondering if the way of presenting this huge data set in the creative way that you've discovered helps you communicate with legislators more effectively. Do you have any experience communicating directly with legislators, and how's their response?
So we actually did the film after we've already had contact with legislators. So that PubChem PFAS tree, I didn't show it in detail here, but one whole section of that was actually working together with Andreas Buser from the BAFU in Switzerland, because he's been looking at the EU regulation, the Stockholm Convention, and looking at how they're defining it and saying, okay, which one do we implement in Switzerland? Should we do the ECHA regulation? Should we do Stockholm? And what is the difference? And so we did a full breakdown of that tree with him, looking at exactly what's written, what is being, what meets the definition, what from our point of view is also what can we not do with the definition, because the regulators write this to make it easy to understand. And then we as cheminformaticians go, we can't actually implement that. It's not possible. So, you know, they'll put wording that makes it vague, but absolutely impossible for us to implement. And then what we've actually done, we've got very detailed breakdown. We can actually also see, okay, which are the new chemicals that meet this that aren't already in your suggested list of chemicals that maybe you should look at. So that's been a really interesting exercise to actually use this as a dialogue. And what he'd done is he'd actually grabbed my slides online and saw it and reached out to us and said, hey, I want to discuss because I think this could really fit in. So one of the other projects that we've got funding with 0PM, it's actually, we've got industry involved in it, we've got also psychologists involved. How do you get people to move away from chemicals and have alternatives? And this has been really interesting because we have a lot of outreach, but also a lot of workshops with the regulators to get this happening. And like, for instance, H&M, who have like moved completely from PFAS, and like telling their story, what does this actually mean for you as a company to implement these alternatives? So it's really interesting, actually.
Thank you very much. Do we have one last question? May I ask something, actually? So lots of your research is based on searching in the huge databases, and one way of looking at it is just reducing the size of the database. But they will grow, I imagine, and probably also exponentially. So what's your, from the cheminformatics point of view, what's your take? What's the future of that line of research?
It's mixed, so we actually, I didn't have time to say it. We have one student who's looking on the exposomics, and what she's been doing in her PhD is, does it help if we get very small disease-specific lists? Does it help if we go PubChem Lite because it has all the disease information in there? Is it better if we go to the entirety of PubChem? You know, she's looking at the different software and how this helps with the annotation, and a lot of this actually depends on your question. So a lot of the work has been, okay, in future, would we do it this way or that way? What we've noticed for the disease, it totally depends on the disease. So we had Parkinson's and Alzheimer's. You would think they're relatively similar. For Alzheimer's, the database we got from PubChem was 40,000. This is too big. For Parkinson's, we had a couple of hundred. So it's a completely different strategy to identify in the chemicals. So that's part of our research, I would say. That's the cheminformatics side.
Thank you very much. So let's thank Emma again. And it's my pleasure to present our next speaker, Laurel McGrain from Fimbrion Therapeutics. So Laurel graduated with a Bachelor in Mathematics from Lindenwood University, and then a PhD in Organic Chemistry from the University of Missouri St. Louis. Then she did a postdoc in Washington University School of Medicine, after which she joined Fimbrion, where she climbed the ranks from senior scientist to director of chemistry. And we welcome Lauren on stage.
Hello. Oh, yes. Hi. First of all, thanks to everybody at the HIP Symposium for inviting me to come all this way. And so I put a little graphic up here. Let's see, go back. Is this the pointer? Aha. Yeah, so I'm from Missouri in the United States, which I figure most of you don't know, but it's in the middle of the country. And I guess despite not being a coastal city, it's actually a pretty cool city. So I wanted to just show a little bit about what goes on in St. Louis. So for one, we have one of the largest med schools in the country. Is this like... I point that way. We have one of the largest med schools in the country. So the Washington University School of Medicine, oh, okay. So sorry, my laser pointer was, I'm just going to put it down for a second. So we have, one of the best med schools in the country. It's hooked to Barnes Jewish Hospital, and it makes it a really prolific area for technology to happen. Our company, Fimbrion, is located just down the street, and it was spun out of technology that was developed at WashU. So 2 WashU professors actually started our company. And if anybody has heard of the St. Louis Arch, it's always there in the skyline. We also have this really cool Forest Park area, and that is WashU. So we're butted up right next to this massive park that hosts a bunch of recreational activities. In addition, the nation's top zoo, and it's a free zoo, as well as a science center and natural or art history museum. I also heard that Germans like beer, and so we are host to the Anheuser-Busch Budweiser headquarters. And over the last, I would say, 10 to 15 years, we've developed quite a craft brewery scene. So we have now over 70 craft breweries in the greater St. Louis area. And if you want to know more about those, you can visit that website, stlbeer.org. Okay, so now that the important stuff is out of the way, I'll tell you about using C-manicides as FIMH antagonists as oral therapeutics for urinary tract infections. So our company began a collaboration in 2016 with GSK to try to develop this technology and bring it to clinic, and we successfully did this. We currently have a compound in phase 1b clinical trials right now, and while I can't disclose the structure today, I will talk about some of the synthetic hurdles and the struggles and the things that we needed to overcome in order to make a manicide into a good oral therapeutic. So just some background on urinary tract infections. It's estimated that there's 400 million cases a year, with 100 million in the US alone, which amounts to a $6 billion cost to the healthcare system. This infection predominantly affects women, with 40 to 60% of all women experiencing a symptomatic UTI in their lifetime, but 12% of men will also experience one, and of these individuals, 30 to 40% will experience recurrence. The main causative pathogen is uropathogenic E. coli, or UPEC, and it's responsible for about 85% of uncomplicated UTIs, so cases of simple bladder cystitis wherein renal function is not impaired. So this does not include more complicated cases that involve the kidneys or catheterization. So all this together, it's a common theme, and it's not really a surprise that antibiotic resistance is on the rise. So here's a little cartoon of the UPEC bug right here, and it has this type 1 pili extending from it, and at the very tip is the carbohydrate lectin-binding FIMH, and it binds the oligomanicides that coat the bladder urothelium. And FIMH is a virulence factor and is absolutely required for UPEC to be able to establish and maintain an infection. So our approach was to develop a small molecule glycomimetic FIMH antagonist that would block UPEC attachment and thereby disrupt the infection cycle. So here's a co-crystal structure of FIMH bound to an endogenous oligomannose 3 ligand, which is an epitope found on high mannose glycoproteins that coat the bladder urothelium. Highlighted in yellow here is the trisaccharide motif that is responsible for the binding interactions. So what you notice about this pocket is there's a very deep polar pocket here, and it's very specific for alpha D-mannose, and the residues are invariant across all strains of UPEC. Surrounding this pocket is a more hydrophobic region, and it's quite flexible, and it can accommodate a bunch of different ligands, so it makes it a really nice target to design for. In contrast, here's our lead biphenyl, our early lead biphenyl manicide, which is bound in the same polar pocket, but then you notice this hydrophobic region called the tyrosine gate is in a much different configuration. And so what's driving the potency with our molecules is a pi stacking interaction here with tyrosine 48, and then that's supported by this electrostatic interaction with arginine 98. So for our early structure, we wanted to focus on optimizing the potency as well as some of the drug-like properties. So before I talk about potency, I just tell you, or not, maybe I won't tell you. Okay, I just want to tell you about our potency assay. So we use something called the hemagglutination inhibition assay. And it's a functional cellular assay that does track really well with SPR-KD, but it gives us a much bigger dynamic range to work with to rank our compounds. And so this assay works that FIMH-mediated hemagglutination occurs between guinea pig red blood cells and E. coli, and the EC90 value just measures the concentration of mannocide needed to inhibit 90% of the hemagglutination. And so for reference, D-mannose has an EC90 value of 1.5 millimolar, and our early lead was already at 1 micromolar. So going forward, our plan was to optimize the regions highlighted. We first wanted to explore ester group replacements to improve the metabolic stability and hopefully improve the potency. We wanted to look at this B ring here to try to improve the PK as well as the potency. And then lastly, I'll speak in a few slides as to why we specifically wanted to look at the ortho position of the A ring. And really, this is the only chemistry slide you'll see in the entire talk, but it just shows how simple these things were to initially assemble from commercially available parts. So whatever component you needed, a simple glycosylation reaction and a Suzuki coupling reaction and deprotection, and you'd have your product. Okay, so this is a lot of things, but basically, this is just cherry-picked some of the different analogs we made over 6 years here. And the main storyline is that we were able to turn this methyl ester into the methylamide, and not only did we improve the potency, but we also improved the metabolic stability. And then later we found that if we cyclized that amide into the isoquinoline ring, we got a huge potency boost, as well as improved some of the PK properties like solubility. And so you can just see some comparators scattered around these analogs here. I'm showing a panel of meta-substituted. We also did explore para and ortho, but overwhelmingly our meta-substituents were more active. So I'm just showing you a sampling of those. We were also inspired by the ester and the amide. We explored some di-substituted derivatives where we got a nice potency hit, and then of course some other bi-aryl B rings over here. So we really considered this analog here to be our lead B ring. And so on the next few slides, or scattered throughout the rest of this talk, you're really going to see a lot of this ring, as well as these two earlier leads as we continue to optimize our mannocide. Okay, so here's the ortho A ring. And why did we want to explore this position? Because everybody else did. So all the way back in 1987, it was reported that if you put a small hydrophobic group there, you would get a potency boost with your inhibitor. And so, of course, we're going to try the same thing. And it turns out that was absolutely true with our analogs as well. And if we look at the crystal structure, where the ortho-substituted analog is in green, we see two reasons for the potency increase. Number one is there's a small hydrophobic pocket here that this group was able to access to increase the binding interactions. Further, in accessing this pocket, it changed the torsional angle of the biphenyl group and aligned the B ring for better pi stacking. All right, so if we strictly look at potency, then you would look and say, okay, this CF3 group here, although I know it's bad, it's good we didn't use it. So this CF3 group here looks fantastic for potency. But if we look at the urine PK, since our target is in the bladder, if we look at the urine PK, what we see is that it was absolutely terrible with respect to urinary exposure. And so looking at this graph, we repeatedly found that the methyl substituent gave us the best combination of potency and urinary exposure. I also want to point out the y-axis here and how different it is between the plasma and the urine. you can see that the values are not even reaching 1 micromolar in the blood, whereas they're reaching upwards of 10 or 100 micromolar in the urine. And so these things are getting cleared very, very rapidly from the blood into the urine, as well as metabolized. And so if we look at the corresponding PK data for these two compounds, it's not really a surprise that the oral bioavailability of these compounds is so low. And we can also look and see that the half-life is pretty terrible. In fact, the values were so terrible we couldn't measure any oral values for that compound. So of course we wanted to see how our advanced lead compared to these two analogs. And so if we look at the PK data for this, you can see that we did improve the oral bioavailability, presumably by making a more soluble B ring here. It's more distributed in the tissues. But when we looked at the urinary exposure for this compound, it's pretty similar to the other compounds. So it's very similar to this 4284 here. But it wasn't all bad news because if we look at the potencies of these compounds, they are in the nanomolar range, right? And our concentrations in the urine are in the micromolar range. So we should still expect to see efficacy, even though the PK is not where we want it to be. And so here's an early chronic model that we had. So we let an infection develop over 2 weeks, then gave a single dose of our manicide, and then sacked the mouse 6 hours later. And what we can see is that all of the compounds did show a greater than 3 log drop in CFU, which is greater than 99% bacterial clearance, with the best analog being the 5254, isoquinolone. And so at this point, it was kind of 6 years of optimization, and we weren't really happy with where we were at. We felt like we should have a more potent compound. And I think we kind of addressed the elephant in the room, so to speak, in that we knew that if we wanted more efficacy, we were going to have to solve the problem of the blood instability. And so we turned to the fact that the O linkage was a metabolic disaster. Not only is it acid labile, but it also gets degraded very quickly in the blood by enzymes as well. And so we sought to look for different linker alternatives to improve the blood PK. And so here's just a sampling of some of the different that I made to explore this. So we have an amine linked and a thiol linked and a methylene linked. Those were all about 2 to 4-fold less potent. We did the C-linked and the N-linked amide as well as the triazole, and those were so bad that I don't even know what the values were. Then we explored the S and the R hydroxymethylene linkers. And excitingly, we found that with this enantiomer, we got a 2 to 4-fold increase in potency depending on the analog, so 31 nanomolar from 62. But interestingly, if we use the other enantiomer, we got a massive decrease in potency. And if we look at the docking structure, what we see is that which was also later confirmed by crystal structure, is that there's a water-mediated hydrogen bond here that the R enantiomer can access that the S enantiomer cannot. And in addition, these torsional angles change again, and now this B ring is almost perfectly aligned with the tyrosine 48. So it's really increasing those binding interactions. And so we have this linker. It should be more stable now, it's more potent, so then, moment of truth, is this going to help our urinary exposure? And if we look at the C-manicide in green here, we can see that now we're getting a more sustained release of manicide into the blood over time. So this was fantastic, and we started putting this linker on everything. Of course, our lead isoquinolone ring, we have the linker on this as well, and we got a nice 4-fold potency increase. This is not an error. We actually saturated that ring, taking the benzyl groups off. And so on the remainder of the slides are going to be unpublished data that we did with GSK. And we started by comparing the efficacy of this really potent C-manicide here with this much less potent O-manicide. And so if we first look at the urinary exposure of two compounds, we now have it normalized for potency. And so what you see here is the y-axis is now fold over EC90, and I think it's a much better way to represent the urinary exposure. It helps you interpret the PK/PD much easier. And if we look at the C-manicide here, you can see that it has therapeutic, at least it's over its EC90 value for at least 12 hours and hits maybe a Cmax of around 300-fold over its EC90 value, whereas 4269 only reaches about 80-fold over its EC90 value. What's also really cool in this experiment is that we also basically, I don't know what 500 mg per kg looks like, probably a paste, but we gave the mice a massive dose of D-mannose just to see, because there's kind of mixed reports out there of women taking up to 3 grams of mannose a day out of desperation. And you can see that they don't know anything about oral bioavailability, that over 24 hours, at no point did the D-mannose even reach 1.5 millimolar, so it's just not even absorbing into the system. So this is a more recent developed model that we did with GSK, where it's the prophylactic prevention model. So we treated with mannocide half an hour before infecting and then sacked the mouse 6 hours later. And what we see in this efficacy is that, as expected, we don't see any statistical difference in dosing the mice with 500 mg per kg mannose relative to the untreated vehicle. But what's somewhat unfortunate is that we also don't really notice a difference between our really potent compound and our much less potent compound. They're pretty similar in about a 2.5 log drop of CFU. And some of you may have noticed I didn't really dwell on it too much, but the urinary exposure for this C-manicide, given that it's 8 times more potent, should have been much higher on this graph. So what I kind of failed to mention is that we actually took the oral bioavailability the wrong direction. We did solve the problem of instability, but we introduced another problem, is that now this stable molecule doesn't want to absorb. So again, we are back to solving how to improve oral bioavailability round 2. The permeability assays, Keiko, things like that, absolutely did not work for any of our compounds. They just all came back as red and terrible. And so we weren't really learning anything from these from those assays. And so we started digging through all of the physicochemical properties and looking at all these things that are supposed to make up Lipinski's rules and whoever's rules of how to design a good oral drug. And what we actually found is that properties related to lipophilicity strongly influenced the oral bioavailability. And over time, we were actually able to come up with an equation that did predict whether or not a mannocide of ours would have good oral bioavailability. But I can't share the equation with you, but I can at least sort of give you an example of the things we were looking at. So if we look at the polar surface area of the O-mannocides, you can see that it's much lower polar surface area than these C-mannocides. And if we look at the log D, or the distribution coefficient of the O-mannocides, we see that it's much higher, meaning it's much greasier. So the short version of what's going on here is that we just need to increase the lipophilicity in order to improve the membrane permeability. And so we figured at this point we really had 2 approaches. We unfortunately can't do anything about the most polar part of the compound that must stay intact. So we either needed to revisit our idea of what was a good linker here and try to find something more nonpolar, or we could revisit the idea of exploring more nonpolar B rings, understanding that we were probably going to take a potency hit in doing so. And what's really cool is we had massive success in both approaches, but for the interest of time and what I'm allowed to talk about, the last 3 slides, I'm just going to show you our success using approach 2 here. So these were the very 2 first lipophilic C-mannocides. I should probably just say lipophilic rings. But these lipophilic mannocides were our very first attempts at sort of seeing if we were on the right track with this approach. And if we look at the potency data here, as expected, we took quite a hit with respect to potency. But what we did do is slightly improve the polar surface area, and we really did bring that log D value up. And what this translated to was a massive boost in oral bioavailability. So I mean, I guess 9 and 17 doesn't sound great, but when you're coming from 0, it was absolutely fantastic. And since the project had started, this was the highest value we'd ever seen for amanicide. What also really inspiring is that maybe some of these values aren't as bad as they seem, artificially low in rodents. So in other species, we actually found there was much more variance in the oral bioavailabilities. So that gave us hope as well that maybe we were on the right track and things would look better once we got it in humans. So, okay, we took a potency hit, but if we look now at this urine PK, what we see is that instead of being at 300-fold for Cmax, we're now 3,000-fold over the EC90 value. So surely, at this point, this should translate to an improvement in efficacy despite the hit in potency. And that's exactly what we see. So now we have these 2 lipophilic manicides here, and they're giving the same drop in CFU at 10 mg per kg that these manicides here were giving at 25 mg per kg. So it's been a huge increase now in efficacy. And so we took this more potent compound 2 here and also did, what I didn't mention is this is still the prophylactic model, but we also did a model wherein we infected twice prior to dosing with the manicide and then dosed 3 times, 6 hours apart, and then sacked the mouse. And what we see is a really nice dose response here, and it shows efficacy at doses even as low as 0.05 mg per kg. And at 5 mg per kg, you see about a 2-log drop in CFU, which is equivalent to macrobid and just slightly inferior to the other 2 antibiotics here. But again, this was only our very, very first attempt at doing this, so this really showed we were on the right track and really inspired how we then looked and tried to develop for our clinical candidate, and that led to the discovery of our candidate 388-2347. And with that, I would like to acknowledge people. So Carbex started GSK $7.5 million to fund the phase 1 clinical trial. So thanks to Carbex. And the phase 1B clinical trial is hopefully wrapping up soon, and we do actively continue to support the clinical trials by doing research, biology research in our lab. And then I would like to thank everybody at our company here. The people in the purple directly worked on the project, and the people in pink here, Jim, Mike, Catherine, and Eugene are the ones responsible for slowly, and without my knowledge, turning me from a synthetic chemist into a med chemist. And then I would like to thank Zenfu for starting the project at WashU. Earlier I showed you a zoomed out location of where Fimbrion was, but this is our building. It's an incubator space where a lot of companies are at, and that's hosted by BioSTL and BioGenerator. This is what the inside looks like, and we're in the back corner here, and that's what our lab looks like. So, thank you.
Are there any questions to Laurel? Yeah, over there.
Laura, thank you very much for the inspiring talk, and certainly for showing us the complexity of bringing something like this from the lead status to a clinical candidate. I have 2 questions. So one, you already alluded to prophylactic treatment, because this is going to be an adhesion inhibitor, right? So, but you also showed therapeutic data.
Yes.
So what is the phase 1 study? Will that be a prophylactic study?
It's treatment.
It's treatment.
Yeah.
Okay.
Yeah. So, but the thing is that the binding of the bacteria to the bladder is not a static event. So if the bacteria just bound and never let go, the bladder lining is sloughing off constantly, and you would lose those bacteria. So they've evolved over time to be able to let go during flow conditions and then reattach. So just because you don't prevent the initial attachment, there's lots of opportunities throughout the infection cycle. So, and these disrupt biofilms as well, so.
I have one more question, if possible.
Yes, please.
Yeah. Maybe a little bit mean question to a chemist, but does your group investigate into the resistance mechanisms? Do you know anything about the resistance occurring against the FIMH?
Yeah, I mean, yeah, so that's, I was having a conversation with somebody earlier trying to get through the hurdles. Anti-infective group at GSK was helping us co-develop this, and they want resistance profiles and things like that, and we couldn't come up with any because we're not killing the bacteria, right? We're just keeping it from binding, and it just happily gets flushed out. So there, as far as we know, we don't really have. You know, you can mutate the pocket a little bit, but seeing as the binding pocket binds endogenous mannose, I don't know how much the pocket could actually mutate, and then it wouldn't be able to bind your bladder either. So. Yeah.
Have any more questions? Anna.
Thanks, Laurel. So I was wondering, you showed some docking figures and also some crystal structures. Can you tell us in how far this optimization program was structure guided, or did the structures come after the fact, so to speak?
No, we got a structure really, really early on. There was, in fact, I think a manicide all the way back in 2000 was serendipitously found in a crystal of FIMH. So I think just they were just trying to crystallize FIMH, and whatever media they were using had a butyl or heptyl manicide in it. So I mean, we had crystal information long before. So it was nice. It was actually really nice. I missed that.
Great, thank you.
Yeah.
Right. One last question, and then we
I'll go back to the side. I don't know.
Postpone the rest of the discussion to the coffee break.
Thank you. Just a quick question. Because you optimized for potency first and then found poor oral bioavailability, and then you came up with an equation to perhaps monitor that, do you think it's possible to optimize for two things, potency and bioavailability, at the same time in one sort of process?
I mean, ideally, that's that's what you want to do. It really took GSK's funds and their resources. The problem was, as our group was just focused on the potency, and we didn't want to venture into non-potent compounds, and we also didn't have the money or the means to put things into animals and see how they did. So you're dealing with calculated properties. And then here's the other thing. Some of our compounds that were less than 1% oral bioavailability, they were clearing 99% of the bacteria. So we knew it was working, but yeah, that, you know, it's working. Help us get that last little bit of oral bioavailability that we need. So, yeah.
Thank you very much.
Thank you.
Thank you very much for a great talk. And it's my pleasure to introduce the last speaker of this session, Markus Lang of the University of Halle-Wittenberg.
Okay. Okay, perfect. This is working. First of all, thank you so much for this opportunity. I highly appreciate that I'm able to speak after those great talks here. So today, my name is Markus, first of all, and I'm a PhD student at the Martin Luther University in Halle. And me and my working group, we are on the hunt for new compounds that show activity against mycobacteria. And today I want to talk about the compound class of AAPs, who specifically inhibit the RNA polymerase of those bacteria. And first of all, I want to start with, why do we need new compounds against those bacteria? So, of course, everybody knows, most infamous, it's Mycobacterium tuberculosis. That is the causative agent of TB, and in 2022, it caused 1.3 million deaths, which makes it the deadliest bacterial seed that we know. If you get a disease like that, you need extensive multi-drug therapy, a lot of combinations, a lot of different antibiotics that you have to take for several months with a lot of side effects. So to have an alternative for resistant MTB is definitely a good thing to have, but there is another group of mycobacteria that is flying under the radar at the moment, and this is the group of NTM, non-tuberculous mycobacteria. They usually manifest as TB-like pulmonary infections, but not everybody can get those infections. You have to meet certain conditions as a patient. So usually immunocompromised patients, for example, organ transplant patients, or patients with structural lung diseases. On top of that list is definitely the patients with cystic fibrosis get those kind of infections. And if we're looking at the species who infect those patients, is most of the time the M. abscessus complex or the M. avium complex. The bad thing about those NTM is that they're inherently more resistant than MTB is, and this one is already resistant, right? Pyrazinamide and isoniazid, they just do not work as standard therapeutics, and also rifamycins, these kind of bacteria, they do get resistances really fast through several different mechanisms. And so that we do not lose that valuable target of RNAPs that rifamycins inhibit as well, we need new substances that inhibit RNAP against mycobacteria. Exactly, and this is when the AAP compound class could come in handy. So the compound class was first found in a high-throughput screen when GSK released a compound library in 2013, and it showed activity against MTB. The best thing about this compound class is that the target site on RNAP is actually different from the ones that rifamycins have, so there is a low probability of cross-resistance, and we can inhibit strains that are resistant to rifamycins. So first of all, we were interested in the in vitro activity of this particular hit compound, and against MTB, we see MIC90 values of 1.7 micromolar, so already pretty good. M. abscessus shows a value of around about 12 micromolar, so much more resistant also to this compound class, and M. avium promising 3 micromolar. And this is also true for other NTMs. So there is a huge panel that causes these kind of diseases, and it's generally active against these NTM as well. So next, we checked on the bactericidality of this compound class, and we were quite happy to see that within a concentration range of 4 times its MIC90, we were able to kill 99% of the bacteria. In that case, M. abscessus, which is a really good property of new drug candidates. So we can call the compound and the compound class itself bactericidal. Okay, and then we did that in our labs. We did a fluorescence-based macrophage infection model, and we developed that because NTM are able to infect the macrophages and the immune cells of their hosts to evade the immune system itself. And as you can see, at 12.5 micromolar, the hit compound is able to completely inhibit the growth of Mycobacterium abscessus inside the macrophages while they are happily growing at 1% DMSO as a control. And we also see a nice dose response in that regard. So really promising features for this compound class. So and after we saw that, we wanted to synthesize new AAP analogs that are more active and are more drug-like. And we synthesized a whole set of new compounds. And as you can see, that compound class is quite modular. You have that phenylalanine in R configuration at the R enantiomer as a central part, and at the left and right-hand side you have 2 more aromatic systems connected by amides and that aliphatic ring system, in that case a morpholine. And in order to do that a little bit more rationally, we used an MTA RNAP crystal structure that was co-crystallized with a prototype of that AAP compound class, and we were also generating an M. abscessus homology model based on that MTB model to make compounds that are a little bit more tailored to the NTM. And we did in silico molecular docking to see if the compound ideas that we generated that way have actually improved target binding. And that way, we were able to generate a whole set of new compounds over several years now. Not every single compound was active, but one particular substitution got our attention, and this is the substitution of the aliphatic morpholine to their respective thiomorpholine derivatives, and especially their respective oxidated products, so the sulfoxide and the sulfone, because they have really interesting properties. If we look at the in vitro activity of these analogs, we see that a sole exchange of the morpholine to the thiomorpholine doesn't result in better activity. Actually, it's getting a little bit worse against MTB. But if we oxidize the sulfur in these compounds, we see a significant improvement. So, for example, if you compare the sulfone to the hit compound, we see a 10-fold increase in M. abscessus activity, and this is also true for MTB and M. intracellulare as a representative of the M. avium complex. Yes, so the sulfone and also the sulfoxides give us a much better activity of this compound class, and the sulfoxides also give us a better aqueous solubility, which could come in handy. Okay, but there is one problem with this compound class, and this is the insufficient stability. We were already able to incorporate sufficient plasma stability, especially in the analogs, but we see a fast degradation in microsomal suspensions in vitro. And this is why we're currently identifying the metabolites that form inside those microsomal suspensions under the influence of the participating enzymes. And this is really preliminary data, but what we see happens is, on the one hand side, an amide hydrolysis of that pretty efficient analyte bond here, so we get that free carboxylic acid. But what seems to happen much faster is an hydroxylation of that morpholine moiety, here, and a subsequent dealkylation of the alkyl groups. So this is the real problem at the moment, and so we try to develop, after we were able to raise the activity, we now try to develop an analogs that have better PKs with that compound class. And one idea that we had in order to do that, especially when it comes to the analyte bond, we were trying to incorporate small substituents in close vicinity, the ortho position, to that analyte to sterically shield the analyte from the enzymes in the microsomal suspensions in order to prevent the hydrolysis. And with, for example, the methyl group or a fluorine substituent in close vicinity, we were indeed able to ramp up the stability in the microsomal suspensions, but that's certainly not enough, but even that small change comes at a cost, and this is the activity. So if we introduce that small methyl group, we lose activity completely to over 15 micromolar. Fluorine is still tolerated, but we are not seeing the nice activities anymore that we had before. Okay, so this kind of validates that the analyte bond seems to be a problem, but we now know that the morpholine group, the alkyl groups in this particular compound class are the bigger problem. Okay, so as a summary, in the past we were able to get detailed SAR information on this compound class for M. abscessus and also for M. tuberculosis, and we were able to lower the MIC90 value into nanomolar range. But we're still facing that problem of low bioavailability, oral bioavailability, and also the low microsomal stability as an in vitro parameter. So for our future plans, we want to dive deeper into the biochemical characterization of this compound class. We want to perform enzyme inhibition assays, and we also want to generate new cryo-EM structures together with our substances to get new ideas on how to change this compound class. And this is also a plan of us. We want to isosterically replace that morpholine aniline group here and the analyte bond itself, so that this compound class gets more stable. And yes, this was it for the short talk. I just want to acknowledge some people. This is my working group at the University of Halle. I want to thank especially Professor Peter Imming and Dr. Adrian Richter, my supervisors for my project. I want to thank the working group of Thomas Dick at the Center for Discovery and Innovation in Nutley. They are doing the MTB essays for us, as we do not have the proper lab for that. And if you ever need a huge library of mycobacteria, this is the people that you should ask. And I also want to thank Professor Elizabeth Campbell and Barbara Bosch and Mira at the Rockefeller University, who are helping us a lot with the generation of new cryo-EM structures. Yes, exactly. The funding of this project is done by the German Cystic Fibrosis Foundation and the German Scientific Foundation, which we're really thankful for. And we also have some posters, poster 6 and 43, if you're interested in our topics. And yes, that's it. And I thank you for your attention. And if you have any questions, I at least try to answer them.
Thank you very much for a very clear
you.
presentation and getting us back in time, almost. Do we have any questions? Yes.
Yes.
So you already know my question because I always ask this, but do you think the poor PK properties for an oral delivery system might make this an interesting case for inhalation delivery because the infection is there?
It definitely makes that a good case for inhalative administration. So definitely this could be an idea, and this is an idea. If we do not get it into the bloodstream or the digestive tract, this could definitely enhance the properties or the use of this compound class. So inhalative use is very good. Also, the micro devices that we saw could also be nice for that purpose. Yeah, definitely. But we haven't tried that yet, but we should definitely do that. Thank you.
More? Yeah. Please, go on.
Thank you so much for the nice talk.
Thank you.
I have two questions. One is the spectrum. Is this limited to mycobacteria?
This is limited to mycobacteria. It's not effective against any other bacteria like something. There is a sister compound class, the CBR compound class, and this one also inhibits the RNA polymerase of, for example, other bacteria, but they are specifically antimycobacterial.
And the second question is, what struck me was the intracellular activity. That's very nice to have, particularly for mycobacteria.
Mm-hmm.
Does this compare well with known antibiotics against the mycobacteria? Do you know this by chance?
If it's comparable to other
no, whether the 12 micromolar is competitive to rifamycin or isoniazide.
Especially rifamycins are even more active, so they are inhibiting the growth inside of the macrophages at even lower concentrations. This was just, I just picked out a nice concentration that looked good on the pictures, but it's inhibiting even a little bit lower. But rifamycins, for example, are better in that regard.
Thank you.
Right, so if there are no more questions, let's thank Marcus again and all the speakers of this session. Now, since we are almost on time, I suggest we start the next session on time at 1:15. Is it? I don't see anyone from the Orga team who is against it. So now we have a coffee break. Thank you very much and enjoy your break. Attention, attention. It's about time for the last session. What? Klingelt, klingelt. So please, everybody take his or her seat. And I warmly welcome everybody to the last session, dedicated to natural product chemistry, biochemistry, and fancy biosynthetic pathways. And before I introduce our next speaker, I would kindly remind everybody to fill out now the online form for the poster vote, so for the Publicum's prize. So you can take your time until 4 o'clock. Every vote counts, so you can also make multiple choices if you cannot decide. But it would be nice if you would have at the end a very nice Gauss curve to get one clear decision. So please take the time to vote while I introduce Mo, a very dear friend of mine. Mo is actually Iranian, and he grew up partially in Germany. And then he moved on to Brandeis University in the US for his undergraduate studies, where he developed basically his passion for biochemistry and novel enzymology. He moved on and stayed in the US to move on to MIT, and then later on to Harvard. So he stayed most of his time then in Boston, where he worked with really famous people, including also very famous natural product chemists and chemical ecologists like John Clardy and Roberto Coulter, having a Novartis fellowship. And clear path, the next choice was to become independent, an assistant professor at Princeton, where he still is, despite the many offers of different universities trying to catch him. But I think he persisted and stayed in Princeton, which seems to be a good choice, I would say. Mo has published tons of papers on novel chemistry, novel biochemistry, microbial interactions, symbiotic interactions, so all the variety we have and the beauty of it. And that also is reflected in the many awards, which I will not count now because they're all here on my list, but there are many of them. And I think that also speaks for itself. So I warmly welcome Mo now on the stage so that he can present part of his research, I assume. Thank you.
Okay, thank you very much, Tina, for the very kind, very generous introduction. Actually, much more generous than I deserve. Thanks. It's great to be in Germany. I consider it a home, and it's always great to come here visit. And I want to thank Rolf and Tina for the invitation to visit you here in Saarbrücken. Okay, so I want to start with a very simple question. Where do antibiotics come from? I think the public perception is always along the lines of this picture. So the chemists synthesizing antibiotics in the lab from scratch. But of course, with all the nice infectious disease research going on here, this audience knows that the vast majority of antibiotics actually come from microbial sources. In fact, more than 3/4 of all FDA-approved antibiotics are from soil-derived bacteria, such as these streptomyces shown here. So clearly, chemists play an important role in this process, but when it comes to antibiotics, bacteria are the stars of the show. And of course, the history of natural product antibiotics goes all the way back to Alexander Fleming, when he noticed that a fungal species was contaminating his staph plate and generating a zone of growth inhibition. This is sort of a reenactment of his experiment. Here's an original figure from his publication in 1929. He showed that a fungal extract was able to inhibit the growth of some bacteria, not others. He showed that this compound was safe to some human cells, and he called this compound penicillin. Now, we heard a very nice talk by Chris on penicillin and penicillin biosynthesis. Interestingly, the structure of penicillin remained unknown for another 20 years. A number of notable scientists, like Edward Abraham, Chris also mentioned, and R.B. Woodward, believed it contained this strained beta-lactam fused structure. Other notable scientists, like Robert Robinson of the Robinson annulation, believed it was this thiazolidine oxazolone type structure, and the debate was ultimately settled by Dorothy Crawford Hodgkin when she determined the crystal structure of penicillin and showed that it indeed contained this strained fused structure. And I promise I'm not copying Chris's slides. Now, what's interesting in this whole story is that Princeton actually played a major role because the paper by Dorothy Hodgkin was published by Princeton University Press. But even before all of this, Florian Chain had figured out how to mass produce penicillin, and ultimately penicillin was deployed in 1945, coinciding with D-Day, in fact. And this started a real trend in natural product discovery and antibiotics discovery. Selman Waksman was the person who sort of did this in a systematic manner and found a number of novel antibiotics, perhaps the most important of these, streptomycin, shown here, the first antitubercular antibiotic. A lot of his antibiotics came from soil streptomyces, and he would say things that sounded very biblical, like, out of the earth shall come thy salvation. Turns out he was just making these up. These are not from the Bible. Other antibiotics he found at the time, investigators found at the time, were vancomycin and erythromycin, and both of these have been important infectious disease agents, anti-infectives as well. Okay, so if we look at a timeline of deployment of the antibiotic and resistance, what we would see is that there was initially a major period of activity between the 1940s and the 1960s where a lot of the drugs that we use today were identified. This was followed by sort of a quiet period where we still introduced new antibiotics, but these were semi-synthetic in nature, altering existing scaffolds and reintroducing them back in the clinic. Now, of course, I've not listed all of the 160 FDA-approved antibiotics on this chart, but if I did, two aspects would remain. There's been resistance to every antibiotic that's been under significant clinical use. And this innovation gap would also remain, this period where no new scaffolds, antibiotic scaffolds, were introduced. So why is it that we went away from something that was working so well in this 20-year period and entered a sort of a innovation gap period? It turns out the true novelty of the molecules that we found decreased precipitously over time. This is sort of a measure of the chemical novelty of the natural products that were identified, quantified by the Tanimoto factor. And you can see sort of immediately around the 1950s, the degree of novelty of these molecules has gone down steadily. The number of molecules that we found has increased, plateauing at around 1,500 a year or so. But the true novelty of these compounds has decreased since the early 50s. And so what this has led to is not only divestment from natural products as a source of drug molecules, but also, for economic reasons and other reasons as well, divestment from antibiotic research by many of the large pharmaceutical companies. And so what this has left us with are these notorious escape pathogens that we've heard several talks about, or urgent threat pathogens, that are now very difficult to treat and are multi-drug resistant as well. So I guess the question should be, where will antibiotics come from in the future? And we've heard a number of nice talks on this topic. I'll introduce maybe another source that could play a role in this. Right around the time that a lot of big pharma divested from antibiotic research, next-generation genome sequencing methodologies were developed and took off. And these led to the accumulation of now hundreds of thousands of fully sequenced microbial genome sequences. And what these have shown is that the natural products we've identified so far are really just the tip of the iceberg, that there's a large swath of biosynthetic pathways, biosynthetic gene clusters that we cannot access with common or conventional methods, and that even well-studied bacteria are still an immense source of new antibiotics and new natural products in general if we can figure out how to turn on or activate these biosynthetic gene clusters. Okay, let me elaborate on this topic a little bit. I think the example of erythromycin is one of the ones that really demonstrated this to the scientific community. Here is the structure of erythromycin. I showed it briefly before. This is a natural product that's been used as an antibiotic since the 1950s. We have second and third generation erythromycins like azithromycin or Z-Pak. Even today, this antibiotic is not synthesized, but isolated from large-scale cultures growing this bacterium, Saccopospora erythrae. We know a lot about the producer, we know a lot about the drug, and ultimately researchers in the field figured out the biosynthetic genes that are required for synthesizing this complex molecule. The real surprise came when the Ledley lab determined the genome sequence of this organism, and what it showed was not just the erythromycin gene cluster that we already knew, but another 20 or so biosynthetic gene clusters that had no products associated with them. So here we were growing this strain on massive scales all over the globe for several decades, and we didn't anticipate that this strain had the capacity to make another 20 or so natural products and possible drug leads. These are what we now call silent or cryptic biosynthetic gene clusters. These are gene clusters that we can easily detect in microbial genome sequences, but that are either transcriptionally silent or sparingly expressed under normal growth conditions in the lab. And S. erythrae here is not the exception, it's the norm. If we look at a number of actinomyces genome sequences that have been sequenced, we would find that about 10 to 20% of these biosynthetic gene clusters are constitutively expressed. The vast majority of them are silent or sparingly expressed. And so this is the challenge that we tackled when I started at Princeton. We want to figure out how we can activate these gene clusters in the original producers, what their products might be, and ultimately if they would be useful to us in any way. And so the approach that we wanted was one that would activate these gene clusters in the endogenous producer without any genetic manipulation, because we were ultimately interested in the regulatory aspects of these silent biosynthetic pathways. And so that precluded insertion of active promoters or transplanting these gene clusters in a heterologous host. Instead, what we chose was a forward chemical genetics approach, an approach that we've called high-throughput elicitor screening, where we basically insert a reporter gene into the gene cluster that we're interested in. This gives us a rapid readout for the level of expression. We can now screen hundreds of conditions to see just what it takes to activate the expression of that silent biosynthetic gene cluster. Okay, and so to test this idea, we chose an organism and a gene cluster. We chose Burkholderia thailandensis. Really, we could have picked any, but we chose this organism because it's genetically tractable and it had many silent biosynthetic gene clusters. And in this organism, we chose the malactone or burkholderic acid gene cluster. This is a cryptic virulence factor. It's required for virulence in worm models, but it's not expressed under normal conditions in the lab. And because of its important role in virulence, two groups, the Brady and the Hertwig labs, had worked on it intensively, and its product was known. So this provided a nice sort of proof of concept case for us to see if we can activate its expression in the lab. And so we generated the right proper genetic constructs and carried out high-throughput elicitor screening to see if we could activate it. So here is sort of our first set of results from this study. I'll briefly walk you through it. We have each of these bars representing the level of expression of the silent gene cluster in the presence of a small molecule elicitor from our library. These are shown in groups of 16. We have a positive control, a constitutively active gene with the same reporter, and a negative control. This is our reporter in the absence of any molecules. Now, even without sophisticated analysis, you can see that a number of these conditions increased or enhanced the expression of these gene clusters. One's shown in red, the next best 2 in orange, and then the next best 5 in blue. Now, what was surprising to us was the identity of these elicitors. The best ones turned out to be piperacillin and trimethoprim, both of these clinically used antibiotics. The next best 2 were all members of the cephalosporin group of clinical antibiotics, and all the bars in blue were actually members of the fluoroquinolones, also clinical antibiotics. All of these antibiotics kill Burkholderia thailandensis at high concentrations, but at the low concentrations that we used in the experiment, they had sort of an opposite effect. They stimulated metabolism rather than inhibit growth. And so from a fairly limited screen, we found 9 elicitors that gave us plenty to work with, all of these antibiotics. Now, we could verify that these were indeed turning on this silent biosynthetic gene cluster using qPCR. We could show expression of these genes shaded in blue with the two elicitors, anywhere from 5 to 45-fold. More importantly, and this is where knowing the product of this pathway came in handy, we could do a metabolic profiling and see that piperacillin and trimethoprim indeed enhanced or induced production of this compound. In the control, you see none because this pathway is completely silent. A little difficult to quantify because we're essentially dividing by 0 here, but we estimated at least 15-fold overproduction with piperacillin, at least 150-fold overproduction with trimethoprim. So in this proof of concept case, the methodology worked really well. We then went on to apply it to a number of other gene clusters where the products were not known. In these cases, we can see the bacteria turn fluorescent green as they start to turn on or express this silent biosynthetic gene cluster. And ultimately, we were able to find about 24 novel natural products, cryptic natural products using this approach in a number of different bacteria. And I won't talk about these at the moment. Okay, so the approach we basically developed relied heavily on genetics. We had to do incorporate reporters, sometimes generate knockouts, and this was especially challenging with actinomycetes, even though that provided a nice readout for the entire experiment. But we wanted to increase the throughput of this process, and we started to think, how can we replace this low genetic step? And with the advances in mass spec, we thought perhaps mass spec was a suitable replacement for classical genetics. And what we meant with that was we could perhaps start with a wild-type organism, carry out high-throughput elicitor screening, and then just directly read out the metabolome of each of these wells in response to the elicitors. Now, if we use a traditional HPLC-MS, with sort of your typical 20 to 30-minute run times, this approach would still take about 4 to 6 weeks if we include now data analysis. This would be kind of as slow as genetics. But if we could image these wells using some kind of an imaging mass spec readout, perhaps the approach could be much faster. We were intrigued by this idea. We purchased an imaging mass spectrometer, a laser ablation, ESI-MS. I'm happy to discuss it further. But we got really good at using it, and we can easily read out now 500 conditions in one afternoon. And this has really expedited the workflow. And so once we knew what we were doing, we decided to apply it to a real test case. We picked this Actinomyces, Amycotopsis keratinophila. We chose it because it's basically genetically intractable for us. Also, PCR screening had shown that it has a vancomycin-like biosynthetic gene cluster, the product of which had gone undetected. And so we thought perhaps we can turn it on using our approach. And so we carried out high-throughput elicitor screening, read out the metabolomes using imaging mass spec. I'll quickly show you what the primary data look like. For example, with elicitor 1, you might see this kind of a mass spec profile. Elicitor 2, 3, 4, so on and so forth. We then sort of combine all these mass spec readouts into a 3D plot where we're detecting basically the M over Z and intensity of all the metabolites that are present as a function of the elicitor library. A 3D sort of a whole cell metabolome map. Now, of course, the software to analyze this kind of multidimensional data was not available, and so we, and so we developed our own. It's called the Metabolomics Profiler, or MEDX. It's actually freely available on our group webpage, and I encourage everyone to use it who's looking at multidimensional mass spec data. Okay, so is the vancomycin type compound embedded somewhere in this sort of sea of metabolites? Before I get to that question, I just want to point out that this is basically now a high throughput way of looking at cryptic metabolite biosynthesis, whereas with reporters, you can detect the production of a single pathway at a time. With a imaging mass spec readout, you can detect all cryptic metabolites that are produced in one experiment. And this is sort of a global, an image of the global secondary metabolome of this organism, if you will, as read out by imaging mass spectrometry. Okay, the vancomycin type compound, looking for the chloride isotope, was not available or was not detected in this sort of lower M over Z range. But when we went to a larger M over Z range, as these glycopeptides tend to be large, we were able to see a compound with an M/Z of 1811 that actually contained this chloride isotope. Now, if I take a 2D slice out of this 3D plot, what you would see is that the best elicitor for this M/Z 1811 compound are dihydroergocristine and vincamine. These are both plant-derived alkaloids. Both of these are toxic, but at low doses, again, they're inducing production of this metabolite. We were ultimately able to collect large production cultures of this compound and solve its structure, and it indeed turned out to be a novel glycopeptide vancomycin-like antibiotic that we've termed keratinocycline. Just briefly, it has a peptide backbone, much like vancomycin. It's a 6-mer rather than a 7-mer. It has this really unusual oxazolidinone at the N-terminus, and then it has a number of different sugar decorations relative to vancomycin. Okay, so I told you about the reporter-guided approach for finding these cryptic metabolites, the mass spec-guided approach for finding these cryptic metabolites. Of course, you can also do biological activity assays. So you turn on the sort of cryptic metabolome of an organism, and then you do, for example, antibiotic assays to find cryptic antibiotics. And using these three family of approaches, over the past several years, we've been able to identify more than 150 novel cryptic metabolites, some examples shown here, that are not accessible with conventional methods. These are all compounds that are not produced under normal growth conditions. I won't talk about these in detail. I'll just say that by focusing on this cryptic metabolome, we found a new source of natural products and perhaps a new source of drug compounds. Okay, so ours hasn't been the only method. There's been a lot of focus in this area, and a number of other approaches have been developed by various groups for turning on these silent biosynthetic gene clusters. And to me, this problem is now, for the most part, solved. I think we can access products of silent biosynthetic pathways. And so the question is, where do we go from here? And we've been pursuing 3 different topics that I briefly want to talk to you about. We've started to look at the potential therapeutic utility of cryptic metabolites, ecological functions that these compounds might play, and we've also examined the mechanism of induction by low-dose antibiotics, as all of the elicitors that we found have in fact been growth inhibitory in nature. I won't have time to talk to you about this component of our work. I've listed some references here, and I suspect Lone will talk about this to some extent as well. But I do want to give you sort of a brief vignette of numbers 1 and 2 here, the therapeutic utility and the ecological functions. Okay, so I'll come back to the keratinocycline for the therapeutic utility. When we started to do antimicrobial assays and assays in general, what we noticed was that this compound was highly specific for Clostridium difficile. C. diff is the agent of C. diff colitis. It is an infection that flares up when there's dysbiosis in the gut. And this compound, keratinocycline B, was a potent inhibitor of C. diff. It does not target any other bacteria really, but certainly not commensal or so-called good bacteria in the gut, and it had no toxicity against mammalian cells either. By contrast, vancomycin, which is the founding member of this group of compounds, is a broad-spectrum antibiotic among gram-positive pathogens, and it kills good and bad bacteria alike. And so we start to think about what the differences are between these two, even though their structures are fairly similar, and even though the hydrogen bonding partners are all conserved in keratinocycline for binding the D-ala-D-ala terminus of peptidoglycan cell walls. And so if we carry out binding assays with vancomycin and the D-ala-D-ala terminus, we see that vancomycin binds with good affinity, 2.7 micromolar. This is consistent with literature values. Keratinocycline does not bind the D-ala-D-ala terminus at all, so it clearly has a different mode of action. We've looked at crystal structures, determined a crystal structure of vancomycin, of keratinocycline, and looked at its interaction with the D-ala-D-ala terminus by modeling. So here's the structure of D-ala-D-ala with vancomycin bound, or vancomycin with D-ala-D-ala bound. In addition to these 5 hydrogen bonding donors or partners that are important, the 7th residue is also critical. It sort of comes around and hugs the peptide and keeps it in place. That 7th residue is missing in keratinocycline. Instead, there's an oxazolidinone, and we detect electron pair repulsion between the D-ala-D-ala carbonyl oxygen and the carbonyl oxygen of the oxazolidinone. Okay, so we've shown that it doesn't go into the cells. Clearly, it's acting at the cell surface or at the cell walls. And so we've conducted some assays to see what the target of keratinocycline might be. And so we've conducted these sort of drop-down assays. Because the peptidoglycan is insoluble, we're basically looking for keratinocycline becoming insoluble once it binds. When we have a peptidoglycan preparation, vancomycin is clearly diminished as we increase the ratio of the glycopeptide to the peptidoglycan. Keratinocycline clearly does not, or does a lot to a much lesser degree. When we prepare peptidoglycan samples that now contain wall-teichoic acids, these are polysaccharides that are components of many gram-positive bacteria, we see keratinocycline does in fact bind to wall-teichoic acid containing peptidoglycans. We can also generate preparations that have no proteins at all or have proteins and wall-teichoic acids. And through these kinds of experiments, we've ultimately figured out that keratinocycline targets the WTAs, the wall-teichoic acids in the C. diff cell wall. There's a number of additional experiments we've done, but I'll sort of jump to the model and tell you what we think is going on. It's still, to some extent, early days. But what we think is currently going on is that vancomycin targets the peptidoglycan, which is relatively conserved from gram-positive to gram-positive bacteria. As a result, vancomycin ends up being a fairly broad-spectrum antibiotic. Keratinocycline, on the other hand, targets these wall-teichoic acids, which are actually fairly genus and species-specific. The wall-teichoic acid sequence in C. difficile is actually quite different from the wall-teichoic acid sequence in other Clostridium species. And so for that reason, keratinocycline ends up being a species-specific antibiotic rather than a more general or broader spectrum antibiotic. We can also see that in the significant degree of synergy that we detect between vancomycin and keratinocycline, where inclusion of about half a micromolar of keratinocycline drops the MIC of vancomycin against C. diff by about 8 to 10-fold. And so this is an interesting combination therapy, perhaps, that we're looking to investigate further. I don't know if keratinocycline will end up being a drug or not, but I think it's fair to assume that in the future, potential drugs and actual drugs will come from cryptic metabolites found either using our approach or the several other approaches that have been developed by now. Okay, so let me tell you briefly a little bit about the ecological functions of cryptic metabolites. And for this project, we've ventured into the context of the human microbiome. There was an influential study in 2014 by the Fischbach lab that showed that the microbiome is actually a significant source of natural products. They detected at the time about 3,000 biosynthetic gene clusters. But the molecules have been difficult to come by, really for the same reason that when we grow these organisms in the lab, again, these biosynthetic gene clusters are transcriptionally silent or sparingly expressed. And so we've started to use the Heights approach to see if we can gain access to some of these cryptic metabolites. Okay, so here's a 3D plot that we carried out, 3D metabolome map using the Heights approach with Streptococcus suis. This is a swine pathogen. It's a zoonotic agent. It can cause serious infections in humans as well. And so in this case, we used the human metabolite library as a potential group of elicitors. The metabolome map that we see for this organism is significantly more sparse. It's just not as productive as a Streptomyces or Actinomyces species. But we do see this group of compounds that are induced by vitamin B3 type molecules, so nicotinamide, niacin, and anabasine, which has this pyridine substructure. We've solved the structure of this compound purified from niacin treatment. This compound actually turns out to be a so-called RIP. There's a focus on RIPs here. This is the kind of molecules that Julian knows really well. But basically what's going on here is we have a compound that is essentially a canonical peptide, except for this carbon-oxygen bond that's introduced during the biosynthetic pathway of this compound. We've called it threoglucin A. Here is the biosynthetic gene cluster of 3-glucin A, fairly simple. There's a precursor peptide, a radical SAM metalloenzyme, and a peptidase transporter. And what we think happens is the ribosome generates this precursor peptide. The radical SAM enzyme then, this is a metalloenzyme that links these two unactivated positions, the C-alpha of glutamine and the threonine oxygen here to generate the aliphatic crosslink. And then in the last step, the leader peptides removed to generate the final 3-glucin product. Okay, so that's how it's biosynthesized. In fact, the biosynthesis is fairly diversity-oriented. We find about 20 other 3-glucins that all have this aliphatic ether crosslink, but are either C-terminally or N-terminally truncated relative to 3-glucin A. Okay, so 3-glucin is basically a cryptic metabolite produced by a Streptococcus bacterium, elicited by B3 vitamins. What is its role in the human microbiome? What is it exactly doing for Streptococcus suis? And so we started to do antimicrobial assays and other types of experiments using 3-glucin, and we never came up with any significant biological activity for some kind of a suicide activity, where this compound was just killing the producer. So it didn't kill any other streptococci, other enterococci, other bacteria, fungal cells, human cell lines. All it did was just kill the producer. And so this had us puzzled for a while. This activity was strongly dependent on the aliphatic ether crosslink and a dyad of aromatic amino acids at the C-terminus of this compound family. And so we started to do dose response assays, and what we found was that there were basically two regimes of activity. One that is non-physiological at very high concentrations, where the bacteria no longer recover. And then there is a physiological range in the sort of nanomolar concentration range to low micromolar, where we see bacteriostatic growth inhibition. So growth is delayed, but the bacteria ultimately recover. Okay. And this type of behavior is reminiscent of what's sometimes observed with persistence induction or increased tolerance that's associated with delayed growth or decelerated growth. And so we could show that this behavior persists whether we add the molecule in the mid-exponential phase or right at the beginning of culture. And we could show that high concentrations are cidal, where we see actually a drop in survival, and low concentrations are, in fact, bacteriostatic. Okay, so if this is associated with persistence, it would allow the organism to persist in the face of exogenous threats or exogenous antibiotics. And so we carried out a simple experiment where we grew Streptococcus suis to mid-exponential phase, treated it with 3-glucin or with control, and then subsequently treated the cultures with ciprofloxacin, 200 micromolar. This is about 200-fold above the MIC. And what we found was that 3-glucin was, in fact, significantly protective. It allowed the bacteria to survive 350-fold better than cultures that were not 3-glucin treated. And so it's early days on this project, but the model that we're sort of pursuing further is that 3-glucin is some kind of a dormancy-inducing or persistence-inducing cryptic metabolite that protects Streptococcus suis against exogenous threats and exogenous toxins. And you might imagine that this would be especially helpful in the complex confine of the human microbiome, where microbes are constantly fighting with each other, sort of carrying out chemical warfare, and being able to persist these constant threats would give Streptococcus suis a competitive advantage. Okay, so with that, I'm going to conclude quickly. I think we have learned that bacteria are renewed sources of new natural products and possible therapeutic leads as long as the proper discovery approaches are used. Heights is one of those approaches, but there are other ones. What we have learned is that antibiotics beget antibiotics, and we've repeatedly used old antibiotics to discover, or known antibiotics, I should say, to discover new cryptic ones. And I think the stage is now set to characterize this vast hidden metabolome that we haven't been able to access before and try to see if these molecules will be of use to us. I showed you some of the directions that we're pursuing further. And I hope what I've done is not just show you where antibiotics have come from, but maybe offer an avenue where antibiotics could come from in the future. I want to thank everyone that contributed to this work. I've had a fantastic group of students and postdocs. Everybody that contributed to this project is shown here in orange. I'm thankful to our collaborators for the crystal structure of keratinocycline B, these funding sources, and again to Rolf and Tina for the invitation to speak. And thankful for your attention and happy to take questions.
Thank you so much for this really inspiring talk about all the cross topics we have. So there are certainly questions. Yeah, okay, Julian.
Using the wait, is it off? Okay. Have you tried using the threoglycin as an elicitor on secondary metabolite production in the producer strain? Or in general, have you tried the natural products that you get from the strains to use them as elicitors and see what happens?
Right. So threoglycin itself doesn't trigger anything else. Actually, there's nothing else to encode. There's nothing else encoded in this organism. So it doesn't act as an inducer of any other compounds. The other natural products certainly are, yeah. We have a sort of a library of these 150 cryptic metabolites. We use them for different sort of experiments. But you know, the best elicitors are just antibiotics. And so the library that we screen most commonly now are just low-dose clinical antibiotics. Yeah.
Thanks. Lorenz?
Thanks, Mo. That was terrific. So you sort of fluctuated between talking about silent gene clusters or cryptic gene clusters.
Mm-hmm.
So what, in your opinion, are these truly silent, or do we, I mean, is there transcriptomic data that tells us they are silent, or is it just that we don't know the chemistry of what's coming out of it?
Yeah, so I think those are two different categories. Silent means, as you said, truly transcriptionally silent. Cryptic it just means the product isn't known, and that could be because we're not using the right maybe extraction protocols, maybe the right precursors aren't there, so it's just more, or maybe it's because it's silent. So cryptic is just a broader term. In terms of whether we know that they're silent or not, the estimates vary depending on sort of who does the study, but at least 50% are silent. And sometimes much more than that. So 50 to 90% is what's been reported in the literature, depending on organism and the research group. Yeah.
We have one more question over there.
Hi. Thanks for the fantastic talk. I'm wondering, for the threoglycin, what makes this producer, so if it is a suicide effect, what makes the producer Streptococcus suis so special, comparing with other streptococcus? What's special in the genome, or what's the mechanism?
Yeah, you know, actually, the more we look, the more we found that it's not special. Actually, a lot of streptococci make these bacteriostatic suicide compounds. And the suspicion that we have right now is that a lot of them are using this approach to decelerate growth and to persist. I hope that answers your question.
Thanks a lot. In the interest of time, we continue. I have one question.
Sure.
Oxazolidinone, how it's made, do you know that? Which enzyme introduces the carboxyl group?
Yes, in threoglycin, right?
Yeah.
Yeah. So yeah, we know how that's made. Yeah, it's a radical SAM enzyme. It has 4 iron 4 sulfur cluster. It has 2 of them. One of them activates the threonine hydroxyl. One of them generates a 5' deoxyadenosyl radical, and then you get coupling.
Yeah, so interesting biochemistry.
Yeah, right.
Thanks a lot. So thanks again for the speaker.
Thank you.
So now it's my time to introduce the second speaker, Lone Graham. So she's actually a biotechnologist by training, and now I would say ecophysiology, and now covering a whole broad spectrum of, I summarize it as microbial, chemical, ecologist with biotechnological background, something like that. So you can imagine that she has a very broad spectrum, very broad expertise, and that's also reflected in a long career in academia, also being basically consultant, not only to funding agencies, but also industry. She also got numerous awards and funding. Also, the list is very long, so I have known Lone also a very long time, and she also knows Mo already since like a decade now, I would say, roughly. 15 years now. Wow. So I'm very happy to have her here. She's really an inspiring person in the microbial ecology field and with natural product experience. The stage is yours.
So I think I'm going to try the DTU approach. Anya went over here, and I'm just going to copy her. We come from the same university. Let's see if that works. And I can. So Mo introduced you to what looked like simple questions. Answer might be a bit complicated. I also have a very simple question. So why do bacteria or filamentous fungi or other creatures produce antibiotics? And the danger of being the second last speaker of a fantastic day like today, and I should thank Christine and everybody for inviting me, is that at least the first 10 of my slides, they've been dealt with already. So you can lean back and we can think a little bit of this as repetition. Because I'm also going to start with penicillin, and we've heard about that innumerable times today. I still think this is probably close to the original picture when Alexander Fleming discovered it. I would like, sort of, for the sake of, I don't know, we can talk about gender equality or microbe equality. So here's a filamentous fungi. It's producing a compound that's killing bacteria. But I would like to also to highlight Elizabeth Hazen and Rachel Brown, that from a soil streptomyces, we've also heard a lot about that, discovered a compound that killed a fungus, actually, the Candida. But somehow they never get mentioned in the same way. No matter for that, you saw the figure that Mo brought you. We had the golden era of antibiotic discovery. And I think in many ways, until maybe not so long ago, this is how we have worked with these metabolites, antibiotics, but also other groups of so-called secondary metabolites from microorganisms. We've had the drug focus. And that is no big surprise. I mean, think of how they've revolutionized medicine. However, what we set out to ask we started thinking about it 8 or 10 years ago, that is really these antibiotics that fall under the broad category of secondary metabolites or specialized metabolites or natural products, whatever you want to call them, are they really only secondary? What are their functions in the producer, in microbiomes? And you can pick out from the elicitation approach of Mo that once we take these compounds to sub-inhibitory concentrations, which may be their natural concentration in real life, then they serve a multitude of other functions. And I'm going to take you through a couple of data or stories that I think can illustrate that. I want to throw one thing, and maybe you know this gentleman. I didn't. So Alexander Fleming got the Nobel Prize for discovery of penicillin. He didn't get the Nobel Prize for discovering that a filamentous fungi kills a bacterium. That's attributed to this young French officer or gentleman. Maybe you're all aware of the story, but he actually has a thesis from 1887 where he describes that a mold, a penicillium, actually can kill or inhibit bacteria. But he got left by the Nobel Prize Committee, or they deliberately awarded the award for the chemistry part of it. Anyway, you've heard this. So when I say what are the functions of these molecules, it's of course not true that we don't know. We know that some of them are toxins, some of them are metal chelators, specifically siderophores. Some of them are indeed antibiotics, especially in inhibiting concentrations. So of course we know something about the natural function or the presumed natural function of these molecules. I should say that there's a tendency to thinking of them as sort of weapons of mass destruction, and what I'm going to show you is that that is not always the case. I had a PhD student who very famously in his thesis wrote that primary metabolism is what makes life possible, secondary metabolism is what makes life exciting. He left science. I don't have to go through this. I don't have to go through this figure. In this audience, it's not really necessary, and you've heard endlessly about biosynthetic gene clusters, but it's really the beauty of antibiotics-secondary metabolites that the genetic basis is clustered in these groups and there's a modularity. It's sort of a Lego-like structure. I'm from Denmark. And I really like to quote one of the fathers of anti-smash, Tilman Weber, who's a very close colleague and friend, who says, nature has really been kind to us in organizing it this way. Anyway, what we've done is we work in different kinds of model systems. So I, just like Anja Boysen, I have a so-called Center of Excellence, really nice bag of money for a long time where you can pursue ideas of really sort of curiosity-driven science. So we work in anything from natural systems. This mic is funny. Anyway, we work in natural systems. We started out there, we really wanted to work with real microbiomes, but as you will see as the talk progresses, we've sort of gone back to working with pure cultures. Sometimes you need to reduce stuff in order to understand what's going on. And I'm going to give you 3 examples. I'm going to introduce my favorite bacterium. I know there are people in the audience that knows it quite well. I'm not sure it's their favorite bacterium. But we were introduced to Pheobacter, which is a genera under the marine Roseobacter group bacterium, one of the most common groups of marine bacteria at all in the oceans. And we were approached by a very, very big turbot grower, aquaculture farmer, based in the northern Galicia. And they were culturing turbot. And at the larval stage, they don't have an immune system, which means that they just die of infectious diseases all the time. It's known that it's bacterial infectious diseases because treating them with antibiotic help. Antibiotics we need to take out of the food chain. We've heard that repeatedly also today. So we were approached and said, do you find anything in our natural microbiology within the system of this aquaculture that we can use instead of antibiotics? So the biocontrolled probiotic concept. And we sampled and we tested and we sampled and we tested, and this is sort of Alexander Fleming's assay taken into maybe not high throughput screening, but at least throughput screening, where we basically grow isolates from the environment, we replica plate them onto a lawn of the fish pathogen in question, and then we isolate the organisms that have a clearing rate. That's our Alexander Fleming moment. And we discovered 2 groups of bacterium that could inhibit the fish pathogens. One group was Vibrios. They turned out also to kill the fish larvae, so perhaps not the best probiotic you can think of. But the other group that consistently inhibited the fish pathogens were the Pheobacter or the Roseobacter group bacteria. They grow in beautiful rosettes under certain culture conditions, and we know that the molecule that's the cause of their antimicrobial activity is called tropodithietic acid. It's probably the least complicated secondary metabolite you've seen as a structure on the screen, and I'm not going to make anything more chemically complicated than this. It's a very, very fascinating molecule. I think we could do a full weekday seminar about it, but let's just leave it at that. They're widely distributed, not just the Roseobacter group, but the organisms that produce TDA. We can look at them in the various sequence-based databases that we have from the ocean environment. For instance, the Terra Ocean Database, we've searched for the genes that are encoding the enzymes that are encoding, that are taking care of the production of TDA. And all the green ones on this map, world map, that are sites where microbial DNA, bacterial DNA has been isolated and it's harboring the TDA biosynthetic genes. We were fortunate enough to be allowed on board a global expedition, a research cruise, where we basically approached that we'd like to sail around the world, we'd isolate bacteria from the waters, and we would scavenge the oceans for novel TDA producers. And somebody believed in us. So we were around the world, and all the blue spots is where we have isolated culturable bacteria, where we've actually verified chemically and genetically that they produce TDA. I'm really happy that this happened quite early. It was back in 2006 and 2007, because when we then sort of continued our work, we discovered that if we drove from DTU, which is just north of Copenhagen, and roughly 40 kilometers west to the Roskilde Fjord, where you have the Viking ships, we also found the TDA-producing Roseobacters, but I'm not sure we would have gotten on this trip if we had known that beforehand. We know a lot about the ecology of these organisms, and that's a topic for another talk. But we started out with the aquaculture part of it, and I'm justifying our interest in these organisms. What we've done here is we've taken eggs, cod eggs, and we distribute one egg into a well of a microteeter dish. The egg will hatch, and we will have a tiny, tiny comma-sized larvae. Then we very deliberately infect them with Vibrio angularum, that's a potent fish pathogen, and what you see here is the cumulative mortality during such a trial, and not surprisingly, the ones that, this is funny, the ones that have been infected, we kill all of them, that's the purpose. Even if we don't infect them with anything, there's still quite a significant mortality. These are really vulnerable, tiny, tiny creatures. However, if we at the same time where we add the pathogen, also add a TDA-producing Pheobacter, you can see that there's hardly any mortality at all. So we can completely abolish the mortality and control the pathogen, and the larvae are completely happy being in the presence of the TDA producer. If we knock out the ability to produce TDA, and I should say that in this particular experiment and in a couple of subsequent experiments, we have actually not done the genetics of the TDA knockout, but Mo very kindly at some point in time, I can't remember when, donated a transposon mutant which has a knockout in the so-called TDA-B gene, so that's actually where it comes from. But you can see that the larvae also die, but not to the same extent as the control when we add the non-TDA producing Pheobacter, indicating that other features of the Pheobacter play a role in this microbial interaction. But what does it do apart from that? So we've repeatedly, when we've talked about this probiotic concept, we've been asked, what happens to the rest of the microbiome? You're introducing or reintroducing a very potent antibiotic, mark the quotation marks, organism. Isn't it going to completely destabilize the microbiome? And you can almost hear from the rhetoric what the answer is going to be. So Natalie set up a system which is simple but takes a lot of manual work. So she took an algal culture, so it's in the algal cultures, in the aquaculture that we find these organisms. She added the TDA-producing wild type. She had a non-axenic but non-inoculated culture, so it's a tetraselmis with all the native microbiome. And then we added the mutant that can no longer produce TDA. And then every 7 days she would sort of, so these are photosynthetic algae, they go into a light incubator, and she would sort of re-inoculate them, dilute them, re-inoculate them every 7 days, and she would do that for a number of months. And every now and then she would take samples, count the algae, count the bacteria, take out microbial or extract microbial DNA, so we could look at the microbiome composition using various sequence-based analysis. And first of all, if we look at the overall richness or diversity, if you wish, of these algal microbiomes, you can see the 3 different treatments up here. There's the wild type, there's a mutant, and then there's the axenic, non-axenic control to which we've done something. And there's really not any difference whatsoever. Once in a while, there's a slight statistical significance, but it's really, if you look at the global, it really doesn't matter that we've added this potent antibiotic producer to the system. If we look at statistical comparisons of microbiomes at the different time points and between the different treatments, what we see is that time matters. So that's what you see here. So the dimension that explains most of the change and the differences in the microbiome composition, that is really time, and that's no big surprise. We probably have a bottle effect. But if we look at what we call the system, whether we've added an antibiotic producer or not, it really doesn't matter. It explains almost nothing of the variance in the system. So the answer to that question that we've been getting repeatedly is really microbiome resilience is strong. However, what does happen is that in that algal microbiome, there are other Pheobacters. So there's a native population of Pheobacter, and that is actually what you see in the black line here. And if we start at the bottom, which is the control, then you can see that over time there's a slight increase in the Pheobacter. So it actually proliferates during this algal experiment. However, when we add the wild type that produces TDA, there is no growth possible for the native Pheobacter. So what we see happening here is really a very tight collaboration between 2 very, very closely related species or organisms, very similar amplicant sequence variants, which is the level we distinguish at. So it's really not a universal effect against the microbiome, it's a very targeted effect. Now that led us to take the next step and say, okay, this is an algal microbiome, it's selected by the algal community, but what happens if we take this out into almost real seawater life? And we know that the Pheobacters are very good biofilm formers. We consistently isolate them from biofilms, especially in harbor areas. So Pernille, that was the PhD student on this, took small stainless steel plates. She coated some of them with the wild type of Pheobacter. She coated others with the TDA deficient mutant of Pheobacter, and then she had plates she didn't do anything to. They were allowed to form a biofilm, then she took them out, and then she placed natural seawater around them. Let's see if we could get this to work. Here we go. It's down there now. And again, let time pass, took out these small plates, isolated DNA, and then asked the question, will the presence of a preformed biofilm with this TDA-producing Pheobacter, will that actually cause everybody else to stay away? And again, you can sort of hear from the language what happens, and it's all based on amplicant sequencing. And actually, the difference is not very large on an overall scale. So the most important parameter is time, explaining 30% of the variation we see in the microbiome. The effect of having a TDA producer present in the system explains a little bit of the variation, but it's really not a dominant driving force in the assembly of this microbiome. At least that's when we thought about, that's how we thought about it when we looked overall on the microbiome and the amplicant sequencing. But then we started sort of digging a little bit closer into it, and the question we asked was, are there any genera or species that are preferentially repelled by the TDA-producing organism, or the opposite, potentially allowing them to colonize? And the answer to that is that yes, there is. We actually see that when we've pre-coated these plates with the TDA-producing wild type, there are actually organisms, and that's indicated by the green bars, there are organisms that appear in higher relative abundance than in the control. So much to our surprise, the presence of a TDA producer is not a major repelling force, it is actually attracting something. And we made a very bold statement that we had to discuss with the reviewers quite a bit, that if we look at a molecular network of the species or ASVs that are present in this system, we can see that if we have pre-coated with the mutant, then everything is just one big, I don't know, messy bundle. But if we have pre-coated with the wild type, we see very strong associations around the wild type. So one could almost talk about the TDA producer as a keystone species. We got away with it, but I'm not sure the reviewer was really happy. Let's move, and I'll try and speed up now. I can see the next speaker is coming. We took it to a simpler experiment saying, what happens, how do two antibiotic bacteria coexist? And we picked two that we know they occur in microbial biofilms. One is a Pseudoaltromonas piscicida, and the other is our TDA-producing Pheobacter. And what we noted was that when we grew them as colonies next to one another, the TDA production in Pheobacter, which is indicated by this brownish pigment, which is really a TDA iron complex, was diminished close to the Pseudoaltromonas. Now that could be growth inhibition, which we know it's not. It could be that the Pseudoaltromonas degrade TDA, or it could be that the Pseudoaltromonas specifically reduces the production of TDA. So we constructed a GFP reporter fusion in one of the TDA biosynthetic genes, and what you see in the upper curve is the TDA production in a Pheobacter growing alone, and you can see that when we have it, let's get it now, when we have it next to the Pseudoaltromonas, and you'll have to trust me, it's up here, we actually see a marked reduction in the expression of that gene and in the biosynthetic pathway as a whole. We actually also have mass spec imaging photos. I didn't bring them because they're so loaded with salt crystals that you almost have to be religious to believe that the chemical data shows exactly the same, but they really do. So Peter, who's the PhD student doing this, he took the two cultures to a liquid co-culture so we could do qPCR, RT-qPCR, and he demonstrated that indeed when we have these organisms in co-culture, the expression of the TDA genes is much reduced compared to the monoculture, and it's a very specific effect. We have them at the same CFU and the same growth densities. We don't know the molecular mechanism because of that, but that's what we're working on presently. The last story is we think of TDA as an antibiotic molecule, but what does it actually do to the producing cell? Is it only an antibiotic? And again, the red arrow indicates where we're going. So here we actually picked up doing our own genetics. So Laura constructed a scarless deletion mutant in a Pheobacter. It's a Pheobacter piscinae that comes from algal culture. And what we noted, not surprisingly, was that the mutant has no antibacterial activity. Its growth rate is unaffected, and then lots of other stuff happens, and I'm just going to give you the data with that. So first of all, taking away the ability to produce TDA changes the cell morphology of the organism. The wild type forms long, elongated rosette cells. The mutant is much smaller. Here is a motility assay in a 0.4% agar. The wild type does move, but the mutant moves much, much faster. And remember, I told you they grow at exactly the same growth rate. So it really changes that. The mutant, on the other hand, is not as capable of forming biofilms as is the wild type. So there's something about the motility biofilm, the planktonic biofilm stage, that has been changed. We then went on and thought, okay, that's fine with all the looking at the cells in the microscope, but can we, by combining proteomics and transcriptomics, say something about what's happening? And to some extent we can. I can tell you that the transcriptome and the proteome, where we're comparing the wild type and the mutant, show more or less the same, and we can do nice Venn diagrams. But what I really want to highlight is that what we saw and see is that the mutant, where we've taken away the ability to produce TDA, massively releases a prophage and a so-called gene transfer agent. Gene transfer agents are virus-like particles. So you've probably all heard of transformation as genetic transfer mechanism, horizontal gene transfer. Conjugation is another mechanism. Transduction is another one. Gene transfer agents are really viral particles. The genes are embedded in the genome of the bacterium. They don't encode their own production. It has to take care of that. But they're sort of lifeboats. So they'll sort of randomly grab genomic DNA when the cell is lysing and ship it off to a neighboring organism. So it's sort of a lifeboat ensuring that when the organism is not surviving very well, at least the genetic material of the bacterial cell is transferred to the environment. So here we have an organism, we've taken away the ability to produce TDA. Maybe it makes it a less capable organism. It's not as good as forming biofilms. And maybe then the way that the bacterium ensures that its DNA will survive on a grander ecological scale. Again, we do not at a regulatory level or molecular level know what's going on now yet, but we have a PhD student that's working on it, and I think he's hoping that he will get there before he finishes. So I hope I have convinced you, and I'm not sure you needed convincing, that microbial secondary metabolites are not just secondary. They're produced because they provide a multitude of advantages, not just being defense molecules. We can see that they have many, many more functions: regulations, gene induction, gene suppression. There's lots of data I didn't show you, but they have very specific and very targeted effects. What we don't know is really what's the regulation and what are the molecular mechanisms when these molecules are sensed, not in a killing fashion, but in some of the other fashions. And does that matter to a center or an institute that's interested in drug discovery? I think it does. I think if we understand a little bit more about the ecology and the ecological functions, I think we can also develop different strategies for going out and discovering novel compounds. So it has merit in that particular way. I have a lot of people to thank. Many of the faces up there, not with names, but with faces, are former or present members of the Center of Excellence that has funding from the Danish National Research Foundation. And we have a great collaboration with Tom Gilbert and his center, and received funding from quite a number of other organizations, for which we're, of course, very grateful. And I'm also very grateful for your attention, and I'd be happy to take any questions.
Thank you, Lone, for this fascinating talk, spanning marine natural product chemistry, ecology to probiotics. I think there are questions. No?
Can't see anything.
So clear? Yeah, there's one in front.
So it's a very naive question, if that's okay, from a chemist. Do you have any feel for the extent and mechanisms to which nucleic acids are transferred in a seawater, for example, between cells of the same organism and between different organisms?
In open seawater, not. I would think that in marine biofilms, which is probably the hot spot for exchange of genetic material, I would think it's all of the above. I think basically conjugation, transformation, any of those mechanisms are operational in the transfer of genetic material.
So I guess it is studyable using labeled materials.
I don't think there's a lot done in natural marine systems. So there's a lot of studies of biofilms in sewage plants, for instance, and how antibiotic-resistant genes are transferred one way or the other. But I don't think there's a lot in natural marine systems. I mean, we'll take the organism into the lab, and then you'll study what they can do. We know that bacteriophages are widely distributed, and some of them will be cargoing material.
Generally, the studies are done by modern sequencing methods, but I'd like to see it by radiolabeling.
Yeah. I'll have to say that I really don't know. Maybe there are studies out there on labeled material and how it's transferred. But I honestly don't know of them.
I actually have one question. Did the larvae later on survive? Did you make a probiotic mix that they could sustain?
So the larvae in our experiment were totally happy until day 9. And what you may not have noted was that then they started dying in all the samples. And that was a very embarrassing part of the experiment, because at that point, the larvae have been eating the egg yolk, and they opened the mouth because now they want real food. And because we weren't feeding them, they were dying from starvation. And the point here is that as long as the larvae are feeding on the egg yolk, they're not considered animals. So we don't need animal ethics approval to do the experiments. But the minute they open the mouth, they become animals. So every other experiment we've done since then, we actually stop before they start feeding. But I should say we have exposed Artemia. We've worked with flounder, cod, turbot, sea bass, sea bream, larvae, rotifers. We've exposed every creature on the planet to these TDA producers. We've also done that because it's known from work by Mo and collaborators that some of these Pheobacter can also produce algacidal compounds, and that really blew our aquaculture work a little bit when that article came out. We've not seen any evidence of this having effect when we have them in these kinds of systems.
Thanks a lot. I think there's time to thank again, Lone. Now comes our last speaker before the poster prizes, Robin Hertel from the University of Kiel. And I think he will talk a bit more about biochemistry, format nitrate transporters as potential drug targets. The stage is yours.
Thank you. Yes, I like to present you the format nitrate transporters of Naegleria fowleri as a potential drug target. I am a first-year PhD, but also started this project during my master's thesis. And I like to introduce first PFFNT. This is a lactate transporter we previously described and shows that it is a novel anti-malaria target. And this is a formiate nitrit transporter of Plasmodium falciparum. It's a homopentamer, which you can see to the left. And it could be shown that compounds like BH267 meta, which we developed in our lab, are able to kill the parasite at low nanomolar concentrations. PFFNT is a vital lactate transporter for the parasite, and by inhibition of this transporter, lactate accumulates in the parasite acidificates and swells and finally dies. Here you can see the inhibitor bind to the central pore of the PFFNT, and the newest generation of drug-like compounds against this transporter contains the CN324 compound, which is modeled on the left side into the surrounding amino acids of the transporter. And parasites cultured in red blood cells were treated with these compounds at the Bernhard Nocht Institute, and they show this compound show lower EC50 values than chloroquine, which was previously used as anti-malaria drug, but due to resistances is no longer in use. FNT-type monocarboxylate transporters are prevalent in many unicellular microbes and absent in multicellular organisms like the human. This, together with the results previously shown, makes them very attractive drug targets. So obviously we look if there are other microbes which we can target on this transporter. And already for Toxoplasma gondii and Amoebia histolytica, we find and characterize FNT transporters. And by genome analysis, we also identified potential transporters in Naegleria fowleri. Naegleria fowleri is a free-living amoeba. Natural habitats are warm freshwaters, and if contaminated water gets into the nose, the parasite can enter through the olfactory nerve into the central nerve system, and causing there the primary amoebic meningoencephalitis, also called PAM. And the PAM went lethal in 97% of the cases within a few weeks. And yeah, now via BLAST with known FNTs, we identified 3 potential FNTs in the Naegleria fowleri genome, and obviously asked if these are real transporters which can transport the common substrates like format, acetate, or lactate. But before we started expressing the transporter in Kiel, we decided to send some of our PFFNT inhibitors to Anjan Debnat in California, who tested these inhibitors against the parasite. And yes, some of these compounds kill Naegleria fowleri in very low micromolar concentrations. The 4 best compounds are also shown here in the top. Yeah. I actually started the project with analysis of transcriptomic data out of the NCBA database to look if they are expressed and how big the transcription rates are. I decided now to skip this part so I have enough time to show you that we have here real transporters in Naegleria fowleri. This is a cell acidification assay which is based on accumulation of lactate due to inhibition of the human monocarboxylate transporters. And we can see in the first row that cells which didn't express additional transporters acidified. You can see this by a brighter yellow color by adding these inhibitors which stop the lactate export. In contrast, if we look at PFFNT, no acidification occurs because it can export the lactate, like we can see here. And similar behavior is seen by NF-FNT2, and this strongly suggests that we have a lactate export with NF-FNT2. Also, we added the PFFNT compound BH267 meta, which blocked the PFFNT and the cell acidify again. NF-FNT2 didn't show this behavior. NF-FNT3 acidify also with no inhibitor targeting NF-FNTs, and so in this experimental setup it wasn't able to transport lactate. Now that we have this indirect data showing maybe a transport, tested if we can measure the transport of formaldehyde nitrite transporter substrates directly. So we used radioactive labeled substrates. We may do this in a dictyostelium discoideum system. This is a common model organism in labs known for its ability to form multicellular complexes, but we worked in this unicellular stage, and then we tested the import of 14C labeled format into the dictyostelium cells, and we can see that NF-FNT1 as well as PFFNT show an uptake of format. So obviously my next steps will be to see if bigger substrates like acetate or lactate are also transported by this transporter, and also if the NF-FNT2 and NF-FNT3 is also capable. Yes, at the end, I'd like to thank the team in Kiel surrounding Professor Beit, especially Maura Möller, who started the project during my master thesis. I joined her and now continue the project at Intana Bioscience, where I performed the cell acidification assay, and last but not least, Anjan Debnat, who performed the testing of the PFFNT compounds against Naegleria fowleri.
Thank you very much for this very great talk, inspiring talk to treat this very nasty amoeba. Are there any questions left? I don't see. Everybody's hungry, I guess. Right? Blood sugar level low.
Yes, Robin, thanks for a nice presentation. I do have a question about your cell acidification assay. Would you be so kind to go into detail a bit more?
Maybe. Okay, I don't think I can go back.
Can we have the presentation back, please? It's coming in a second.
Okay, I will go from here. Yeah.
I don't really have a specific question, just could you maybe explain it again, please, because I think it's maybe more interesting than it looks like.
What a comment. So I think the question was how it's done. Or maybe it's also more a point for later on the barbecue, because if it's so interesting, it takes probably longer to explain. Okay, I think we leave it like that, and I would like to thank all the speakers of this session. It was really an amazing session. And I think we go ahead. Don't run away. No, not yet time. So now it's, no, you can run away. That's fine. But don't run too far. So I think now it's time for the poster prizes and the presentations and maybe some concluding remarks.
So it's almost the end of the symposium, but we've had the pleasure of having many very good posters around on display all of today. And we've had a jury that had a very hard job of selecting a total of 4 posters. So first of all, I would like to thank our jury, which is comprised of Brigitta Loretz, Daniel Krug, Martin Emting, and Kenan Bossuyuk. So a big thanks to all of you. And we will now get to see the 4 winners of the 4 poster prizes all at once. And then the idea is that the 4 of you come up to the stage so that you get your prize and we can take a quick photo. So let's see. There we go. So there are 3 poster prizes that were defined by the jury. They go to Helena Pickford for a poster entitled Uptake of a Molecular Cargo by Catecholate Siderophores into E. coli. The second prize goes to Katrin Eisenmenger for her poster entitled Synergistic Activation of Bacterial CLIP-P. The third prize goes to Vincent Libis, I'm sorry. The poster is entitled Scalable Discovery of Microbial Secondary Metabolites through Synthetic Biology. Maybe Vincent had to leave early. Does anyone know Vincent? Is there anyone else from the same group? Oh, very good. Sorry? Excellent. And finally, we have the poster prize that all of you voted for, and this goes to Mario Fares for a poster. There you go.
Yeah, thanks a lot. Thanks a lot.
Yeah, so the prize comes with prize money. There's 100 euros that come from the Förderverein of the HZI, so we should thank the Förderverein for their generous donation. But there's no cash with it. So don't be disappointed if there's no cash with it. There's an instruction how you get the money. So you have to write one email and declare your banking details, and then you will receive the money. All right, so this is our prize jury and the people who received the prizes. All righty, I think it's up to me. Say some, well, it's not finished yet, right? But the scientific part is over. I hope that everybody brought some time to join us for the barbecue. If you don't know how to get to the hips, follow the queue. It's hopefully not raining. And we moved this symposium on purpose from June into May, because the first 12 years of this symposium every year consistently took place in the hottest week of the summer. I'm not kidding you. And if it's 40 degrees outside in this building, it's basically unbearable. But now, for the last 2 years, we seem to have managed very well. So temperature-wise, we are doing very good inside here. And I think it will also be good for the barbecue. Anyway, the scientific part is over. Thanks for coming. I think we've seen a tremendous breadth and depth of science, which is actually hard to get. So I'm always impressed what under the roof of pharmacy can be done. You know, if you think about the interdisciplinarity in a pharmacy school, we've heard from microbial ecology into medicinal chemistry. We've seen talks that dealt with bioinformatics, and bringing this together is, for me at least, the large part of the fun of what we are doing here. And coming back to how Chris opened the lecture series, everybody has to find their place. And I think under this roof, there's a lot of places to find for a scientific career, and I wish everybody the best of luck in doing so. Let me thank the people, the numerous people who helped organizing this meeting. I think they deserve another round of applause. And I'll see you for a hopefully variety of dishes. We've seen variety in science, and if you work in academia, you quickly realize there's an enormous variety in researchers. So I think they are more diverse than bacterial, say, species and families and even suborders. And the trick is getting a good team together. And I really want to thank my team for being such a good team. So let's have some fun at the barbecue.